Any questions before we get rolling today? Alrighty, today is Friday, July 9th, same as it ever was. On to the muscle physiology in lecture, origins, insertions, and actions in lab. A handful of assignments due next week on Monday and Tuesday. Again, this is your big 2-day weekend to study for the exam. You get your 1-day weekend on Wednesday to study because Thursday the 15th is our exam, and then no rest for the weekend. We jump right back in on the 16th and start the nervous system, where we have 3, 6, 7 lectures to learn everything you possibly could ever want to know and more about the nervous system. And then we're done. It's crazy how fast this all goes. So that is our game plan for today. Now, after yesterday's class, I got a couple emails that people really appreciated that we took the time to go through the process of the muscle relaxation to make sure that all of it makes sense. And so I thought it might be worthwhile to do the exact same thing, only for our muscle contraction. Again, obviously, this isn't going to be written as an essay question in this fashion, but I think we could benefit from taking the opportunity to make sure we understand the process of the entire muscle contraction and go through all the steps, make sure we understand the information. So again, no new information here. We're just going to take a chance to synthesize this, and by us, I mean you, going through and helping me to go through the entire process. If this was one entire process, how would it go? So let's do that. We want to contract a muscle. Where does it begin?
Decision in your brain.
Excellent. Now, where this is going to be too, there we go. Excellent. Excellent. As we talked about, we make that decision. Actually, I'll start with the bigger, and then if it's too bad, then we'll make it. All right, we make the decision, and of course, when we make that decision, we produce a neural action potential that spreads down the axon. That neural action potential reaches the synaptic end bulb. And what happens when that neural action potential reaches that synaptic end bulb?
Depolarizes the synaptic end bulb.
Right, depolarizes, and depolarizes to what point?
To threshold, negative 60 millivolts.
Excellent. What's the immediate effect of that depolarization to threshold?
Voltage gated calcium channels open.
Excellent. And of course, when they open, which way does calcium moves?
In.
What is the effect of that calcium moving into the synaptic end bulb?
Increase of calcium in the synaptic end bulb.
Excellent. Which does what?
Triggers the release of acetylcholine by exocytosis out of the synaptic end bulb.
Excellent. Or you could also say into the synaptic cleft. Would be fine as well. Now our acetylcholine's in the synaptic cleft, and what happens now?
It binds to the chemical gated.
Excellent. It diffuses across to the motor end plate. And you are correct. And binds to what specifically?
The chemically gated sodium channels.
Are they really just sodium channels?
Cation channels.
Excellent. Excellent. Diffuse across to chemically gated cation channels. Of course, those channels open. And what happens when those channels open?
A little bit of potassium exits and a lot of sodium enters the motor end plate.
And as we know, the net effect of that is what?
Depolarization of the motor end plate down to threshold, negative 60 millivolts.
Well, but remember, it doesn't have to, and that's really the key to this. If the depolarization is big enough, it is possible that it might not be big enough, right? If every time we released acetylcholine, we wanted that muscle cell to fire, we would just hardwire them together, right? This is a counting game. It's accounting. It's beans. It's variable. It's modifiable. So if the depolarization is big enough, then you have the right idea. We produce big enough, let's say, to reach threshold. Then a muscle action potential is produced at the motor end plate. And of course, from there it spreads down the sarcolemma. What happens next?
Sarcolemma travels down the tubules, transverse tubules.
Excellent. Why is that significant?
Depolarizes the transverse tubules and the terminal cisternae of the triad, the two terminal cisternae and the T tubules.
There you go. Excellent. When our excellent. And when the triad depolarizes, what happens? There we go. Samuel's got it. And of course, which way does calcium move in this case?
With the gradient.
True, but in this
So
case,
out
of what?
Out of the terminal cisternae, or out of the sarcoplasmic reticulum.
And into
Sarcomere.
Yeah, into the cytosol. Right. Basically, it's not leaving the cell; it's just leaving the sarcoplasmic reticulum into the cytosol. So we get an increase in calcium in the cytosol. When that calcium enters the cytosol, what does it do?
Binds to the troponin.
What effect does that have on the troponin?
It changes its shape.
Right. And again, we can say it undergoes a conformational change if we're feeling right fancy, but changes its shape is fine. What happens when troponin changes its shape?
It rotates and turns the tropomyosin off of the binding site of the actin.
Excellent. What happens when that myosin binding site on the actin is exposed?
The myosin heads. How would you say that? The primed myosin heads?
Yeah, that'd be fine. Primed, energized, cocked, however you want to describe it. A primed myosin head. Now, again, let's put an asterisk here. If you wanted to describe what it means to be primed, you can do this here. However, you also know that in this cyclical process, we're going to get to priming again at a different place as well. So we could do it either way, but since we mentioned it, let's go ahead and put it here. How do you prime a myosin head?
Attach ATP, and then it hydrolysis of ATP.
Yeah, and hydrolyzes, or we can just say splits. When it splits the ATP, it stores the ADP, the phosphate, and the energy. And what else happens to that myosin head as it's storing this ATP, ADP, phosphate, and energy? How do we know a myosin head is primed?
It's facing away from the M line.
Jacob's got it. Absolutely. Yep. It points away from the M line. Absolutely. And is pointed away, whoops, from the M line. Excellent. So we can put it there, or we can put it, we'll put an asterisk where else we could have written that as well. So now we've got that prime myosin head. Our binding site is exposed. And what does that prime myosin head do?
Forms a cross bridge.
Forms a cross bridge. And what does that mean? You're absolutely correct.
It releases the energy and performs the power stroke.
Well, you're right. That's what happens next. Absolutely. So it forms a cross bridge, but that cross bridge is where it basically binds to the actin. And you're right. As soon as it binds to the actin, immediately that primed myosin head performs a power stroke. And that power stroke is basically three things. The first is that it pivots towards the M line. All right, so it pivots towards the M line using the energy. When this happens, it pulls on the actin, which pulls on the Z disc. And as a result of that, our sarcomere gets shorter. And we know that sarcomere is connected to the myofibril, is a part of the myofibril, which is connected to the muscle cell, which is connected to the fascicle, which is connected to the bones. And so basically this, as we've talked about, generates our tension. Or again, our force. Either of those would be a fine way of speaking about it. All right. But remember, there's a third thing that happens here as well. While performing, performing the power stroke. At ADP and phosphate, let's go ahead and do this. So the TP binding site is open. Excellent. So now what happens?
A new ATP binds to the myosin head.
And just the act of that does what?
The myosin releases from the actin and breaks the cross bridge.
Then what happens?
ATP splits.
Right. So notice
and is primed.
Here where we wrote about the myosin head, we could just as easily have put it here, right? Now, again, when you're describing this yourself, you don't have to put it in both places, but I'm putting it in both places because it shows where it would be fine in either of those two cases. And of course, what happens next?
The
Cycle.
process repeats.
Yeah, exactly. Well, if the regulatory proteins are still out of the way, then the myosin head forms a new cross bridge, and the process continues. And there you go. Just that simply, we have described the process from where we make the decision to the point where the muscle is moving through space. Now, you can see why we don't have this as an essay question by itself and why we've broken it up into more easily digestible pieces. So let's do that. What's step one again?
Communication at the neuromuscular junction.
Excellent. So step one is communication at the neuromuscular junction. Where does that begin?
Making the decision.
Excellent. And goes till when?
The muscle action potential travels down the sarcolemma.
There we go. Bingo. What's step two?
Muscle action potential spread down the sarcolemma.
Well, no, what's step two called?
Excitation, contraction.
There you go. Excellent. Brian, you have a mental block on that excitation contraction coupling. All right.
All
Excellent.
right.
Well, it's just like the second or third time that you haven't been able to pull that term. I just find it interesting. All right. Excellent. So where does that begin?
Spreads down the sarcolemma.
Yeah, we make the muscle action potential and it spreads down the sarcolemma. And where does it end?
The troponin rotates.
Yeah, basically. There you go. Exactly. And so then, of course, what is step three called?
Contractile cycle.
Exactly. Excellent. Where does that begin?
Troponin rotates.
There you go. And then obviously till the end. So there you go. Just that simply, we have described our longest physiological process. Notice each individual step in and of itself isn't hard or really complicated or really confusing. It's just that there's a lot of them. And that's why we break it up into three more easily digestible pieces. All right. Questions on this? All right. We have spent the first 2 weeks starting with a muscle, breaking it down to its most basic components, and then seeing how these basic components work. Now we can finally start building back up. And that's our job now. So any questions on this before we move forward? All right, like I said, we took 20 minutes here to do this, and there's absolutely no new information here on the board, but I think this is still worthwhile to, again, make sure we understand this concept, make sure you understand what I'm looking for. I don't expect you to memorize this and use exactly these terms or this terminology, but this is the way I want it. I want the detail. I want the immediate steps. So again, hopefully this is useful for you guys to be successful on the exam. All right, so let's start the process of building back up. We know now how to contract a muscle. So what kind of things determine how long that muscle contracts for? What are some of the factors that could influence how long a muscle contracts for?
What activity we're doing?
True. Okay, absolutely. That could affect it. But again, let's think back. Again, I warn you right now, I grew up back east in ancient times, so it was a totally different world back there. And when I was in elementary school and you got in trouble, like I did a couple times, you went up to the principal's office. And at our school, when you went up to the principal's office because you were in trouble, he would make you get down on your knees and he would make you put your arms out. And he would put a textbook in each of your hands.
Hmm.
And you had to hold those textbooks up until he got tired. And if you could hold those textbooks up until he got tired, you got to go back to the classroom. If you couldn't hold them up till he got tired, you got swatted and sent back to the classroom. Right? Now, obviously, are you ever going to be able to hold them up till he got tired? No. So if we were smart kids, we would have just dropped them right away, gotten our spanking and gone back. But that's not what you did. You held them for as long as you possibly could, and then you gave up, and then you got your swatting and got back sent back to the class. At least that's what happened to me, right? So yes, some of it is determined by your activity, but did I want to fail that activity? Did I want to drop those books and get swatted? No. So it isn't necessarily that. So again, think of it more at a molecular level. What is it that actually determines how long a contraction lasts?
Muscle fatigue?
Say again?
Muscle fatigue, how long your muscle can actually.
True. We'll talk about muscle fatigue, and absolutely, Samuel's got one of the factors, as we've talked about, is how much ATP is available. Excellent. ATP is going to be vital for this process. What else did we say was vital for the process to continue?
Calcium.
Yeah, how much calcium is available. And how do we control how much calcium is available? How do I determine how long this contraction of my finger is going to take? Why does it stop?
Because you decide.
Exactly. The decision. However, it's not so much the decision that is actually impacting or changing, turning this contraction on and turning it off, but that decision causes action potentials, which of course, as we know, releases the acetylcholine, right? These are the types of things we're talking about. How long I'm producing my stimulation, right? Which obviously is how much neurotransmitter is released. Or let's go ahead and stick with acetylcholine because that is the specific neurotransmitter. That is going to determine how long those sodium channels are open. That's going to determine how long the cell stays depolarized. That depolarization opens up the voltage gated channels, allowing the calcium to leave the sarcoplasmic reticulum. How much ATP is available. And all of these things are things that are modifiable. We can affect and influence. We have a limited amount of ATP available. We can have some control over how much calcium is going to be able to be released and how much is pumped back up. We have the volition control of trying to stimulate that muscle and get it. All of those types of things have the ability to influence and affect how long that concentration, how long that contraction takes place. And as we know, that contraction causes the sarcomere to get shorter. And we can actually see that in the proteins themselves. Here, we've looked before at that sarcomere in a relaxed state. But now we know with the sliding filament theory that when this muscle contracts, our two Z discs are going to be pulled towards each other. They're going to be pulled towards the midline. And so this has the potential to change the size of our regions, right? Again, these things like the A band, the I band, the H zone, all of these things are the regions of the organelle. Oops. That is the myofibril. And they were an A band, they were an I band, there was an H zone or band, either of those are fine. There's the M line, and there are the Z lines. So tell me how, if at all, these change. So let's actually make that. How do the regions of the organelle change during contraction. Does the A band change during contraction?
No.
No. Right? It is determined by the size or the length of the myosin. And the length of the myosin doesn't change. So there's no change to the A band. But what about the I band?
Yeah.
Yeah, it gets smaller because there is more overlap between the actin and the myosin. So because of that, there's less area of actin that doesn't have myosin in it. So notice we go from this region where we can't even see all of the I band to here where the I band is much, much smaller. What about the H zone?
Yes.
Yeah. What happens to it?
Shrinks. Gets smaller.
For the exact same reason, right? Because there is more overlap between the actin and the myosin. In fact, and let's do this, cheat a little bit here. In fact, if we contract the muscle enough, the actin from the two sides can meet at the middle. And if the actin from the two sides meet in the middle, what happens to the H zone?
It goes away.
Yeah. So it can go away entirely. Excellent. What about the M line? Does it change?
No.
Nope, no change there. Excellent. And that leaves us with the Z lines. Do they change?
No.
Well, sort
Change
of.
position, but
Exactly. The lines don't change themselves. That is true. But what does happen to the Z lines is that they are pulled closer to each other. Right. So again, the Z line itself doesn't change, but its position changes. And there you go. Now notice that's really nice and really easy to see when we're looking at it on this illustration, but it can't really look like that, can it? Well, it turns out it does. Here, as we've talked about, using an electron microscope, we can actually look at the arrangement of the proteins inside of our myofibril. Here, we have this beautiful view of a sarcomere with an electron microscope. We can see the A band that contains the myosin, the I band, which only has the very thin actin in it. We can see the H zone, which is the region that just has myosin in it. Notice the part where myosin and actin overlap is much, much darker. And we can even see the M line at the center. So again, this is an actual electron microscopy view of a myofibril inside a single muscle cell. And we can see that relaxed state where the A band and the I band are fairly similar in their size. But then what we can do is contract a muscle, cut the piece of muscle out, freeze it, slice it, bombard it with electrons. And when we do that, it looks like this. Notice as we talked about, oh, I wonder how good I am at this. Let's see. There's our A band. Ah, there you go. Look at that. I have it lined up. There's our A band. Notice our A band hasn't changed. Our M line hasn't changed. Right? Those are the same. Sort of. Right? But what clearly has changed is our I band. There's so much more overlap that the I band is tiny. The Z discs And again, if we want to make sure, we can go back. Notice they've both significantly moved inward. So again, the size of it, the shape of it hasn't changed, but their relationships to each other have changed. And if we look closely, I'll change to a line here. We can see there's still a tiny little bit to the sides of the M line where the actin isn't fully overlapping with the, even though the picture shows it fully overlapping, there does seem to be a little bit of a lighter region right here on the side. So it might not fully be overlapping just yet, right? But it's close. So again, here with an actual electron microscopy view, we can see those changes in a single sarcomere and how those regions of the organelle change. And again, while we're focusing on those effects in a single sarcomere, that action potential spreads like an electrical current along the sarcomere and down all the transverse tubules, stimulating all the sarcoplasmic reticulum, releasing all of the regulatory proteins, causing all of the myosin and actin to interact, causing all of the sarcomeres to shorten at the same time. So when that muscle contracts, it isn't just one sarcomere, but all the sarcomeres that get shorter. And as we've talked about, these myofibrils are connected to the plasma membrane of the muscle cell. So when our contractile organelles shorten, our muscle cell shortens. And when our muscle cell shortens, it pulls on the connective tissue surrounding it, pulling on the fascicle pulls on the muscle pulls on the bone, and we move through space. Now, when we're moving our body through space, right, we're generating that tension by pulling on the connective tissue to produce that muscle movement. And the total amount of tension you can produce by an entire muscle is going to be determined by the number of muscle cells that are contracting in unison, right? Obviously, the number of muscle cells it takes me to pick up my socks is going to be different from what it takes to pick up my textbook, which is going to be different from my desk, which is going to be different from my automobile. Am I likely to be able to pick up my automobile? Yes,
No.
my wife. Yeah, well, my wife is having a midlife crisis and does have a Mini Cooper. But even though it's a Mini Cooper, I still can't pick it up. Close. I can shake it a little bit, but I'm not going to be able to get it off the ground because I'm limited by the total amount of muscles. So obviously, I'm using different numbers of muscle cells depending on the activity. But what about the muscle cell itself? Does a single muscle cell itself has a set amount of tension that that muscle cell produces?
Yes.
Well, it does have a maximum, but do I always have to contract it at its maximum?
No.
No. And that's the key. I can vary how much tension, and that's the key. Notice now, up to this point, we've been talking at things at the level of a sarcomere. Now we're moving up. We're at the level of a single muscle cell. And with a single muscle cell, we have the ability to change how much tension that muscle cell produces. And it turns out we can do that two ways. The first is by the stretch of that muscle cell prior to the contraction, the length of the muscle cell before the contraction begins. And the second is by the amount of stimulation and the frequency of that stimulation, right? If I need to pick even something as light as my phone up here off of the table, I'm not sending one neural action potential there to do the whole act of bringing this up to my face so that I can talk into it. So these are the characteristics that can change the tension in a single muscle cell. Let's start with stretch because I think it's easier to understand. Tension of a single muscle cell can be modified by the length of the cell prior to contraction. So the question becomes why? What actually determines the amount of tension produced? What do you think actually determines how much tension a muscle cell produces? We're playing tug of war, and all of me is on one side and all of you is on the other. Who's winning?
We are.
Why?
There's a lot more of us than you.
Excellent, right? So we've all got our hands on the rope and we're all pulling on that rope, and you guys got more hands, right? And that's basically what happens here, right?
The zone of overlap?
I'm sorry?
The zone of overlap?
Yeah, exactly. That's a key part of it. You've absolutely got the right idea. You've taken the next step in what we're talking about, but you're absolutely right. When it comes to the amount of tension, basically the tension is determined, or let's say it this way. If you don't like tension, let's think of it in terms of force. The force produced is determined by the number of myosin heads that can form cross bridges and perform power strokes. And of course, what determines how many can do this? As Heike pointed out, one of the key factors is the amount of overlap. Let's look at a really simple example. Here is my myosin. And let's say, for argument's sake, that this myosin, I'm not going to draw the big circle heads, but we'll just put the arms. Of the myosin heads coming off it. So I'll just draw my myosin heads like this instead of putting the big bulbous ends on it. All right. So there's my myosin, and my myosin has 12 myosin heads. Okay. Then, of course, I have my actin and my Z disc. And right now, with my sarcomere at this length, how many of my myosin heads are going to be able to grab onto actin and produce a power stroke?
8.
Yeah. 1, 2, 3, 4, 5, 6, 7, 8. So I get 8 power strokes worth of tension from the muscle if the sarcomere is at this length when I start. But what happens if I stretch the muscle out a little bit? We know our muscle has elasticity, so it can be stretched without damaging it. And if I stretch it out a little bit, and then I start a contraction, what's going to happen to the strength of the contraction that I'm going to be able to produce here?
It's going to be weaker.
Yeah. In fact, it's going to be 50% weaker, because how many myosin heads are going to be able to grab and pull here?
4.
Yeah, just 4. And not so theoretically, if I were to stretch the muscle out far enough so that the myosin and the actin don't overlap at all, how much tension can I produce here? Yeah, Samuel's got it. None. Now, you may not have ever thought of this before in your entire life, but you're aware of it. because it's either happened to you or someone you loved, or you've just watched it occur while you're at a bar. There you are hanging out at a bar. Someone's had a few too many Long Island iced teas, gets a little rowdy, and the bouncer comes, right, to break up the fight or whatever's going on and to subdue the rowdy individual. And one of the things they often do to those rowdy individuals is they take their arms and they pull them behind their back, right? Because when you've stretched out the arm, by stretching out those muscles, you weaken how much force they can produce. And now you're able, by keeping the person's arm behind them, you can subdue someone much stronger than you by stretching out their muscles so they're not going to be able to produce as efficient of a contraction. Right? So you stretch out the muscle and the muscle gets weaker. Now notice if we increase the overlap from where we originally started, now how many of my myosin heads can grab and pull?
All 12.
Yeah, all 12 of them. I'm going to be able to produce a very powerful contraction. But remember, as we talked about, our actin, and actually I have to do this. From the two sides, remember, can actually meet in the middle where our myosin line is. We have that myosin in the middle. And our actin can interact with the myosin, interact with the actin from the other side. Notice as this starts to happen, plenty of myosin heads are going to be able to grab onto the actin, but are they going to be able to perform successful power strokes? Are they going to be able to move the actin and pull the sarcomere shorter?
No.
No. And so once again, as we shorten the muscles are going to get weaker again. Which brings us back to the bouncers at the bar. If they can't pull the person's arms way behind them, then what do? They bear hug them and they get their arms tucked in close. Because if you get their arms tucked in close, again, you limit their ability to produce force. So as Jacob pointed out, it's not just the amount of overlap. The amount of overlap is absolutely important. But remember, it's not just overlap, but also we need to have the space for the power strokes. So it isn't all overlap, but we also have to have space for power strokes. Now I've done a really simplistic illustration of this. Your book's got a little bit of a nicer one. Here we see this graph from your textbook that shows this. Notice the area where we have the greatest tension that can be produced is where there's a large amount of overlap, but plenty of space to perform power strokes. Notice as the actin gets closer and closer, they start to interact more, fewer power strokes can be made. And then notice once they hit the M line, and that M line really messes with things, we get a dramatic drop in the amount of tension until it's overlapping so much, the sarcomere can't get any more shorter, and our tension is 0. And then also, the more we stretch it out, the fewer myosin heads overlap with actin, the fewer power strokes we can perform. Plenty of room for power strokes, just not enough myosin heads. Until again, we get to the point where there's no overlap. And if there's no overlap, no power stroke can be formed and our tension becomes 0. Now notice again, there is a normal range for most muscles. And as it turns out, most, there's that pesky word most, but not all, but most muscles in anatomical position are at their optimal length. We'll learn about a muscle today that isn't when we're going through the lab part, but when you're in anatomical position, most of the muscles in your body are at their optimal length to produce their optimal amount of tension. All right, questions on that? Of our two factors, I think this one is the easier one to understand because this one's all mechanical. Can you grab the handle? Can you pull? Right? The more hands you can get on the rope and the more you can pull on that rope, the more power you're going to be able to perform. If you can't get enough hands on the rope, then you become weaker as a group. And if you can get all your hands, but there's no room for you to pull, that's not going to be very efficient as well. All right. Excellent. It is a little bit early, but this is a good natural stopping point. And we don't have as much lecture today, but we do have a lot more lab, although we are on schedule with our lab, which I'm totally thrilled about. But I still think this is a good point to take our first break. So let's go ahead and take our first break. We will restart at 9:02. And from there, we will pick up and see the effect that our nervous system and those neural action potentials have on producing our muscle contractions and how that can affect it. So I will restart the recording at that point. And I will see you guys in 15 minutes. All righty. So we left off and we were talking about how to modify the tension within a single muscle cell. We're still at the level of a single muscle cell. And one of the factors we talked about was length. How long the sarcomere is prior to the contraction beginning. We did all of that. The second component has to do with how frequently or how rapidly we stimulate the muscle cell. Now, with muscle cells, as we said, it can be modified, but there isn't a tremendous amount of modification, and we need a starting point. The starting point, the general rule for a muscle cell is that one neural action potential produces one muscle action potential, which produces one, oops, that's not how you spell produces, which produces one twitch. Now, we know a little bit about the neural action potentials. We know a little bit about the muscle action potentials. What we haven't talked about yet is a twitch. We define a twitch as a single stimulus, contraction, and relaxation of a muscle fiber. Oops, hold on. Didn't mean to do that. Oh, all over the place. Oh, well, guess it doesn't matter anymore. So, single twitch is a single stimulus, contraction, and relaxation sequence in a muscle fiber. Here we see some examples of twitches. Every muscle cell twitches, but they may twitch at slightly different rates. Some will be really fast, like for instance, the muscles that control our eye. Our soleus, which helps us to stand upright, has a very long twitch. So these twitches can go anywhere between 7 and 100 milliseconds. On average, they're about 40. And regardless of the speed, all three of these twitches have the exact same stages to them. Now, I want to move this to the whiteboard and look at this. So let's do this real quickly. So again, I want to start with our rule. One neural action potential produces one muscle action potential, produces one twitch, and a twitch is a single stimulus, contraction, and relaxation. So we're going to draw a graph. And anytime we draw a graph, we always want to label what's going on it. Down here, we have our time. It's a little big. That works. And over here on the y-axis, we have tension. Now, this is a good starting point. What I want to show you, and I'll draw it here first, and then we'll go back. Actually, I lied. We'll go back to the picture and look at it first. Notice again, we have tension here on the y-axis. We have time here in milliseconds on the x-axis. But notice something interesting. Normally, when you put a time of 0, it's right up against the y-axis. But here it's offset a little bit. So let's do that on our graph as well. And I'm going to exaggerate mine a little bit. So I will put it here. And this is time 0. So what does that 0 indicate? Well, that 0 actually indicates when our muscle action potential is produced. That is what our 0 time indicates. Now, as we know, when it comes to the events of getting our muscle to contract, and we went through that whole long process at the beginning of class today, was the muscle action potential the very first event?
No.
No, there was a whole bunch of stuff that has to happen first. We have to make the decision. We have to produce a neural action potential. We have to release acetylcholine. We have to open cation channels. We have to depolarize the motor end plate. And then ultimately at that point, we have to reach threshold. Right? There's all these other events that have to occur before the muscle action potential is produced, and that's why they've offset the 0 time. 0 time is when we get the big event, that irritation of our muscle cell produces the muscle action potential, which, as we just finished talking about, is going to produce our twitch. So since it's the muscle action potential that produces our twitch, the muscle action potential is our 0 point. But they've reminded us that all this other stuff has to happen first. So all of this stuff has to happen first. And that is why they've offset our 0 point on our graph here. All right, does that make sense, hopefully? So there are these things that has to happen before the muscle cell gets stimulated. And that's why they've offset the 0. Now, once we generate that muscle action potential, as we talked about, our twitch has 3 stages. And one of the important things to remember about these stages, how we tell the stages apart, is how the tension changes during the stages. So that is kind of how we define the stages, is by how the tension changes. Now, as we look at our graph, and let's go ahead and do this and do that, we generate that muscle action potential. Now, we know tension is going to be produced by myosin heads grabbing onto the actin and pulling. And the instant that a muscle action potential is produced, are myosin heads instantly pulling on actin?
No.
No, we know that muscle action potential has to spread down the sarcolemma. It has to spread down the transverse tubule. It has to depolarize the sarcoplasmic reticulum. It has to open up voltage-gated calcium channels. Calcium has to leave the sarcoplasmic reticulum into the cytosol and find a troponin and bind to the troponin and rotate the tropomyosin. There's all these events that have to occur first before we can start performing power strokes. So actually, let's go back and make this a little bigger. The first step of our, and again, I'm exaggerating this here for my drawing. The first stage of our twitch is what is called the latent stage. Notice during this latent stage, it isn't that nothing is occurring, but remember, we identify the stages by how the tension changes. And how does the tension change during the latent stage?
Not at all.
It doesn't. There's no change in tension. But that doesn't mean that there's nothing happening, right? Things occur. As we talked about, we have to open our voltage-gated calcium channels. We need to release the calcium. We have to move the regulatory proteins. If you think about it, in a way, this is kind of excitation-contraction coupling. All those events we talked about in excitation-contraction coupling are kind of occurring in this latent phase. And this latent phase takes like, I don't know, 1 to 3 milliseconds or something like that. It's a very, very short period of time. But there is a brief period of time where that is occurring. Then, finally, regulatory proteins start to move. And myosin heads that are primed can grab and perform power strokes. Now, do all the myosin heads all grab at once and all pull at once so we instantly reach our maximum tension?
No.
No. First, a couple are going to grab, and then a couple more, and a couple more, and a couple more, and a couple more, till all of them that are grabbing and pulling are pulling. And this is our second phase, what we control the contractile phase. In the contractile phase, our tension is increasing. And as we just finished mentioning, the reason for this is we have an increase in calcium in the cytosol. So as a result of that, more regulatory proteins are moved. And if more regulatory proteins are moved, we can perform more power strokes. And the more power strokes we produce, the more our tension increases. Now, we have this stage. We're releasing that calcium. We're moving the regulatory proteins. The myosin heads are grabbing and pull. But remember, we just produced one muscle action potential. And is one muscle action potential going to last for a very long period of time?
No.
No. So are we going to be able to keep this cell depolarized for a long period of time? No. And as it starts to depolarize, as we know, our voltage-gated calcium channels close. No new calcium is released into the cytosol. Now, when that occurs, do all the regulatory proteins go back into place and all of the tension goes away instantaneously?
No.
No, we know we have to pump the calcium out back into the sarcoplasmic reticulum. But as we pump it out, less calcium is available, less regulatory proteins are going to be moving, and fewer myosin heads can grab and pull. So what's going to happen to the amount of tension I can produce?
Going to decrease.
It's going to decrease, exactly, until it finally reaches 0. This is what is called the relaxation phase, or the relaxation stage. Don't be confused by this. It is relaxation, but does that mean tension is equal to 0?
No.
No. During the relaxation stage, tension is decreasing, right? There is a decrease in the calcium in the cytosol. So fewer regulatory proteins move. And as a result of that, fewer power strokes are performed. And because of that, tension goes back down. Once tension equals 0, then the muscle cell is at rest. So when the tension is 0 after the twitch, it's back to rest. Relaxation isn't when it's 0. Rest is when it's at 0.
Is the muscle always at a state of like readiness? I mean, helping with like support and.
Yeah, again, like we've talked about, your legs may not be moving right now, but is there plenty of ATP floating around inside the cells? So those myosin heads have grabbed them and they're primed and ready to go. And all it needs is a little bit of calcium to move those things out of the way so that you can start your contraction. Yeah, absolutely. So our myosin heads are ready to do their work as soon as we move those regulatory proteins out of the way. So this is a single twitch, end of story, right? Excellent. All right, we've done it here. Let's look at it, the pretty pictures from your textbook. They've yanked one out. Again, it doesn't matter which one it is. I think it was the gastrocnemius because it goes to about 40. But again, we have that latent period or latent phase where, again, the key is tension. No change in tension during the latent period, during the latent phase. But it doesn't mean that nothing's going on. This is basically when excitation contraction coupling is occurring, right? We're depolarizing the muscle cell, opening the voltage gated calcium channels, releasing calcium to move the regulatory proteins. And like I said, this only takes about 1 to 3 milliseconds. That leads to the contraction phase, the contraction period, where tension increases. As more calcium becomes available, more regulatory proteins can be moved, more myosin heads can grab and pull, and the tension goes up. Again, we only had one stimulus though, only one muscle action potential was produced, and that doesn't last for a very long period of time, so the cell doesn't stay depolarized for a very long period of time. And very quickly it repolarizes. When it repolarizes, the cell doesn't instantly go back to 0 tension. Yes, those voltage gated calcium channels close and no new calcium is released, but we still have the calcium in the cytosol that needs to be pumped away. And that takes a little bit of time. So during this period of time, calcium is removed from the cytosol. With less calcium, fewer regulatory proteins can be moved. With fewer regulatory proteins being moved, fewer myosin heads can grab and pull, and our tension goes down. So latent period, tension stays the same. Contractile period, tension increases. Relaxation period, tension decreases. Back to rest. All right. Questions on that? All right, excellent. So far, so good. gets a little bit trickier from here. So let's go back to our drawing. Here is a single twitch because, as we just finished talking about, one neural action potential produces one muscle action potential, produces one twitch. So if I ask you to describe a twitch, this is how you would answer that essay question. But like I just finished saying, this is for a single twitch. And if you think about it, is that how we're going to move our body through space? Right? A contraction that lasts 40 milliseconds. Is that going to be an easy way to bring my coffee to my face in the morning? No. So obviously, we want to produce many neural action potentials, to produce many muscle action potentials, to produce many twitches. But we have a problem. And here's the problem. That problem is that when a muscle cell produces an action potential, and this is way too big. When a muscle cell produces an action potential, for a brief period of time, it loses its irritability. Remember, irritability means that it is able to respond to a stimulus. When this muscle cell produces an action potential, immediately after that, there is a brief period of time where the muscle cell loses its irritability. And if it loses its irritability, it cannot produce a second, right? It's already produced one, but it cannot produce a second muscle action potential. Doesn't matter what you do to it. As we know, typically to get this cell to fire an action potential, we've got to get it to threshold. We only need a 10 millivolt change. Well, you can take this muscle cell and hit it with a lightning bolt, plug it into the light socket. Whatever you do to this, there is nothing you can do to this muscle cell to get it to fire a second action potential. This period of time where it loses its irritability is what we call the refractory period. Again, notice this refractory period has nothing to do with the first twitch. Nothing to do with the first twitch. If I ask you to describe a twitch, you do not have to talk about the refractory period. The refractory period only determines when we can produce a second action potential. And the good news is this refractory period isn't very long. In our skeletal muscle cells, it's basically, oh, I don't know, 1 to 5 milliseconds. So it's very, very quickly. Notice, is it even done with its first twitch when I can stimulate it and give it a second twitch?
No.
No. So this is before the first twitch ends. All right, so this is before the first twitch ends. So it's not a long period of time, but it is an important period of time. Because what that means is it can't instantaneously be stimulated. You can't bang, bang right in a row and instantaneously get 2 action potentials. There is an upper limit to how quickly we can stimulate it. And that's really what this refractory does. It determines the upper limit on how fast we can stimulate a muscle cell. All right. So obviously the refractory period is related to a twitch, but isn't actually a part of the twitch. It's when we can produce a second one. All right. Does this make sense? It only gets worse from here. So if this doesn't make sense, you need to let me know now. All right, excellent. So we now know some properties of our muscle cell. We know our muscle cell can produce a twitch. We know that there is an upper limit on how fast we can produce a second twitch. But it turns out there are a couple other important properties, or we can say characteristics, of skeletal muscle cells. The next of these is a characteristic known as Trappy. All right, let's draw our graph again. I'm going to change the scale a little bit of my graph. All right. Brian, is that high enough where you can see the bottom of the graph?
Negative.
There?
That's good.
I'm not going to bother writing time or anything underneath it, so that's fine there. I am going to change the scale slightly, though. All right. So again, here we have we stimulate the muscle. We generate that muscle action potential. And we know as a result of that, we are going to get a twitch. And we know that twitch starts first with a latent period where there's no change in tension, and then we get an increase in pressure and a decrease in pressure. All right, I knew I'd draw a little bit under there, but that wasn't intentional. It doesn't obviously go below zero tension, but you get the idea. All right. No new information, we're comfortable with that. Let's say we wait 5 minutes. And 5 minutes later, we stimulate the muscle cell again, generate a muscle action potential. And of course, that muscle action potential is going to produce a twitch. And how do you think this twitch will relate to the first one we produced 5 minutes ago?
Identical.
Yeah, it's going to be identical. Now, I don't know what my abilities to draw identical twitches here will be, but let's pretend that's kind of horrible, but we get the idea. The point is that these two twitches are going to be identical. So 5 minutes later, it's identical to the first. So far, not so bad, right? Excellent. But do we actually have to wait 5 minutes to stimulate it again?
No.
No. So what if we stimulate this muscle cell again immediately after it reaches zero tension? So what we do here, and actually I'll write this one up here. We stimulate a muscle cell immediately after it returns to rest. And again, here's the important thing of this. What is the tension in the muscle cell when it reaches that resting state?
0.
there is no tension left in this muscle cell, all right? The tension is 0. So we immediately stimulate the muscle, immediately after it returns to rate. Tension is 0. So we're not adding a twitch to any tension that's there. There's no tension there. But what occurs is we get a twitch with more tension. So our second twitch in this case is going to be a latent period, a contractile phase, and a relaxation phase. And notice our second twitch is generating more tension than the first one did. We get an increase in tension. Now, the big question becomes, why? What's happened here? Like I said, our tension is 0, so we're not adding tension to what's already there, right? That would make sense. If we were adding tension to what's already there, then sure, of course it would be bigger. But why is it bigger in this case?
Because of the calcium that's still there in the cytosol hasn't been cleared out yet.
Bingo, absolutely. Remember, tension is determined by the number of myosin heads that grab and pull. And what determines how many myosin heads can grab and pull? How many regulatory proteins are moved? And what determines how many regulatory proteins are moved? The amount of calcium. As we've talked about, the calcium isn't pumped away immediately. So what's happening here is that, oh, no, I want that to stay big. I want to come over here and make it a little bit smaller. Even though the tension equals 0, there is still some residual calcium. So when I generate my second twitch, muscle action potential, my second twitch, even though the same amount of calcium is released inside the cell, because of the residual, there is now a little bit more calcium. And if there's more calcium in the cell, well, that's going to equal more tension. We're going to be able to move more regulatory proteins, and we are going to be able to produce more tension. Let's use completely artificial numbers. When we generate a muscle action potential, let's say our muscle cell releases, oops, no, hold on, I want that to stay there.
Either no latent period, right? As well.
I'm sorry?
Would that mean that there's no latent period either?
No, there's still a latent period because we still need time to get that action potential to spread and opening of the voltage gated channels and all that. So let's say that when we generate the muscle action potential, that at the peak, there is 100 calcium that are released, okay? So 100 calcium are released. And when tension equals 0, 90 are put away. Of course, how many are going to be left?
10.
10 left. There you go. You don't even have to take your shoes and socks off, hopefully, to do this kind of math. There is 10 left. So when we generate our next muscle action potential, we now have 110 calcium. And we produce a larger contraction. And then once again, 90 are put away. And if 90 are put away in this case, what happens now?
Left with 20.
Yeah, we have 20 left. So with 20 left, if I stimulate it again right away, notice if I wait 5 minutes, all that residual calcium is going to be put away. But if I stimulate it right away, there's now 20 left, which means that I'm going to have 120 calcium. And if I have 120 calcium, what's going to happen to my twitch?
Bigger.
Yep, even more tension. For some muscles, this process can occur anywhere between 30 and 50 times, where each time it gets bigger and bigger and bigger. Now, eventually, are we going to reach a maximal amount of residual calcium?
Yep.
And that gives us our maximum enhancement for our Trappy. So eventually we're going to reach some point where we're going to reach some maximal tension where every subsequent twitch after that point is going to reach whatever that phase is, whatever that stage is, right? So there is a maximal Trappy tension that can be achieved.
And that maximum tension is based upon the size of the muscle or the number of sarcomeres.
My guess is probably more the size of the cell. The larger the cell, probably the more residual calcium you can have. All right. Again, I got the pretty picture from the textbook. We talked about that, the refractory period already. Here's our Trappy. Again, notice our first one down to 0, up, and then more, and then more, and then more until we reach whatever that maximum tension in Trappy is. The key to this Trappy is that we are not adding waves of tension together. We are not adding tension to what's already there, right? Tension always reaches 0 first before we stimulate it again. What we are getting is residual calcium. That is what's causing this effect. Now, you may have never heard of Trappy, thought of Trappy, right? Been aware of Trappy, heck, even thought of calcium in a muscle cell. But I absolutely positively guarantee you that you know about this concept. Because let's think about it. Right around the corner, allegedly, are going to be the 2020 Olympics. I assume they're still planning on taking place in Japan. Haven't heard any changes yet, but it's coming. And of course, one of the most watched events are the sprints, like Usain Bolt, 100-meter dash, and all those types of things. And those sprinters in track and field, when they're getting ready to get into the stocks and take that 100-yard dash, do they move as slowly as possible to reserve all their energies and save up all of their strength and all of their resources for that burst out of the gate? None of you ever watched a sprinter before? No, they are constantly in motion. They're constantly bouncing around. They're constantly moving. They're constantly taking little steps. And even down into the stocks, as they're getting into the blocks for that takeoff, they're trying to move as much as possible, right? Getting those little bit of movements so that they can build up that residual calcium into their muscles so that when that gun goes off, they can produce the maximal tension contraction using this Trappy to burst out of that gate as rapidly as possible. All right, so if you've ever wondered why those, those sprinters are always bouncing around, right, moving and shaking as they're getting into the blocks, they try to be still for as little as possible, right, up to the point, right when they get ready and go, they're moving because of this Trappy. They're trying to get that built up calcium into their muscles so that they can produce a more powerful contraction when they're coming out of that block to win that race. All right. Questions on that? Excellent. That is indeed an important characteristic of our muscles and this tension that they can produce. However, it isn't super practical for everyday use because as that, and again, no, this isn't my coffee cup, but I don't want to spill stuff all over the place. Is that how I lift my coffee cup? I make faster and faster and faster twitches, stronger and stronger twitches of my arm till finally I get the cup to my face?
No.
No, that's not how we typically move our body through space with a bunch of repetitive, rapid twitches that get stronger and stronger. But that comes back to this concept of Trappy. With this concept of Trappy, as we talk about, we are not adding tensions together. It is always reaching zero first. But here's the trick. Do we actually have to wait for it to reach zero first before we stimulate it?
No.
No. So we can actually add waves of tension together. And when you add waves of tension together, they have an incredibly technical term for it. They call it wave summation. Wave summation, as the name indicates, is when we add waves of tension together. Let's quickly draw ourselves a twitch. We stimulate it. When we stimulate it, we get a twitch, latent period, contractile phase, relaxation phase. And as we talked about, the average is about 40 milliseconds for that. So if we want to add waves together, we must stimulate the muscle before the tension reaches zero, right? If the tension reaches zero, then there's nothing for us to add the wave to. So obviously we have to, want the line, there we go. All right, we have to do it, whoops. We have to do it before tension equals zero, which, like I said, is about 40 millivolts on average. Some muscles it's faster, some it's slower. So again, we'll put that down there so it's not in the way. Obviously, we have to do it before it reaches zero. However, as we've talked about, is there an upper limit to how quickly we can stimulate it?
Yeah.
Yeah. So we have to wait till we have to stimulate it before it reaches zero tension, but we also have to wait until after what?
Refractory.
There you go. After the refractory period. Remember, if we try to stimulate it before the refractory period is over, we don't get a result. So we have to wait till after the refractory period. But remind me again, how long did I say the refractory period lasted for?
1 to 5 milliseconds.
Remember, the latent period itself is 1 to 3. So even with a 1 millisecond latent period, we're still talking about something in the range of just as it's starting to produce tension, somewhere in this range. So if we stimulate this anywhere in between these two ends, after the latent period but before it reaches 0, what we're able to do, we stimulate it. And when we stimulate it, we get a latent period. We get a contractile and relaxation phase, and we are able to add it to what we had before. So we're adding these values together. So essentially, we take this and move it up there. And now we've added that tension together. Again, has to be after the refractory period, before the muscle fully relaxes. We stimulate it, and we're adding these twitches to the previous twitches, and we produce our wave summation. And if we go back to our Trappy, if we're adding waves to this, we don't wait till it reaches 0, but we add it to it. We stimulate it, and we stimulate it, and we stimulate it, and we stimulate it, and we stimulate it. We can produce much more tension with wave summation than we can achieve by Trappy alone. So we can produce a lot more tension with wave summation than we can just with Trappy. All right, questions on that? Now, one last question. Is there going to be an upper limit?
Yeah.
Yeah, absolutely. There's going to be an upper limit. And what's interesting about this maximal tension of wave summation is that when we typically contract our muscles, those muscle contractions typically do not reach the maximal. And that maximal tension we call is tetanus. A typical muscle contraction, like the one we see here, is a contraction where we get this rapid undulation of tension that is near the peak but does not reach it. Now that doesn't seem to make a whole lot of sense, because again, as I'm lifting this coffee cup towards my face, is it shaking from all those undulations as I'm trying to pour that coffee into my mouth? So when I lift this cup, why doesn't it shake? Well, let me ask the question this way. How many of you would be willing to get on camera right now and sing your favorite K-pop song for 2 points of extra credit? Just a big K-pop solo for 2 points of extra credit.
If I knew any, I would.
All right. Well, let's say it this way. How many of you would be willing to, if all of you were singing a song together at the same time, would be willing to sing in a choir for 2 points of extra credit? Why would you be more willing to sing in a choir than you would as a solo?
All the attention's not on you.
Exactly, because there are many voices, and when you have many voices, they blend together. They're not going to be as noticeable if they miss a note or squeak or something along those lines. Well, even lifting something as light as my coffee mug, am I just using one muscle cell to do that? No, I'm using a choir of muscle cells. And when I use a choir of muscle cells, they all kind of smooth each other out, right? Again, you may not have thought of it in these terms, but you're aware of it because you're using many muscle cells to lift this coffee cup. But what if you're trying to thread a needle? If you're trying to thread a needle, you're not using so many muscle cells. And now instead of a whole choir, you have a duet or a quartet of muscle cells. And when you just have that, is it more easy to see that vibration of the muscle cells when you do that? Right? How easy is it to thread a needle if it's not something you do regularly?
Very hard.
Yeah. Now, if you practice it and do it all the time, can you get to the point where you're better at smoothing out those actions? Absolutely. When I was in graduate school in preparation for surgeries, before we were allowed to do our own solo surgery, our PI, my instructor, basically had this jar where they had a dye in the bottom of the jar and a piece of string, a suture, and you had to, using forceps in the jar, you had to tie the string around the dye. And until you could do that, you weren't allowed to do your own surgery. So that practicing of that to get that smooth hand so that you're able to make those incisions and, you know, not kill the brain while you're doing it is important. So these things can be practiced. It can be smoothed out. But if it's not something you're doing every day, we tend to see that. The other place you see that is instead of trying to thread a needle, if you're trying to hold up your friend's sofa going up 3 flights of stairs. As that sofa gets heavier and heavier, what starts to happen to your arms as it gets heavier and heavier? Right? You start to shake. The reason you start to shake is because to maintain the tension, you are having to fire all the muscle cells at the same time to try to produce enough tension because you're so near your max. And so as a result of that, you start to see the undulations because you're synchronizing them. Normally, during normal functions, they're asynchronous, but when you're doing that extreme, it is synchronous. Now, again, I say extreme, but lifting that, you know, that sofa for a friend isn't necessarily extreme. It's just really, really hard. Do we hear stories about grandmothers flipping cars off of babies and stuff along those lines? Yeah, you hear stuff about that. And is it possible to actually get a muscle to contract at its maximal tension? Yes. That contraction, that smooth, continuous contraction at the peak level is what we call a complete or a fused tetanus. All right, this one, I think I said it, but I'll say it again just in case. This one's an incomplete or an unfused tetanus. Well, this is a complete tetanus or a fused tetanus. And this is where you produce a smooth, sustained contraction at the peak. Now, why wouldn't you want to do this all the time?
Damage your muscle.
Yeah, as we will talk about, a muscle contraction is a very violent act. And contracting the muscle at its peak tension can be very damaging to it. So if I'm lifting my coffee cup, is it smarter for me to use 3 muscle cells at their absolute peak to lift it up where I could be potentially damaging them? Or do I just use 6 muscle cells in an unfused tetanus where I'm less likely to damage them, right? So yeah, if you are, like I said, in extreme stress, sometimes it can be capable. For your fine motor skills, you can sometimes train this. And again, artificially, right? Again, you go out into the, you know, back 40 to, you know, just hang out and you're urinating and you grab onto that electric fence. What happens when you grab onto that electric fence?
Fracture your arm.
Yeah, it stimulates all of your muscle cells all at the same time, which can not only is that hard because then you can't let go because all of your muscles are firing all at the same time and you're locked into that position, but it also can be very, very damaging. It can tear the muscles, cause severe damage to the muscle, and can even cause bones to break as a result of those powerful contractions. So it's possible to get a muscle to do that, but we typically don't want it to. All right. Questions on that? All right.
I just thought it was epiphany that, like, when you talk about tetanus, I always thought that was what got into your body, not the actual act of it's causing tetanus.
But see, that's why the tetanus toxin is called that, because it causes these complete sustained contractions of the muscles, which, as we mentioned, is very damaging. So yes, tetanus is a toxin, that tetanus toxin that causes the muscles to tetanize. So that's where it gets its name, because it causes these tetanuses of the muscle, causes it to completely get a fused tetanus as a result. Yep. Awesome. Any other questions? All righty, excellent. That is all I wanted to do from a lecture standpoint today. So let's go ahead and close this. And, oh, I know what I needed. Need that again. Perfect. Excellent. All right. That is everything we need to do from a lecture standpoint for today. So what we are going to do now is switch gears to the lab, working through our muscles, origins, insertions, and actions. We left off and we'd worked our way through all of the muscles that we needed to know for the trunk. So from there, we are on to the muscles that move the upper limb, starting first with the bicep brachia. So again, this is where the starting point will be. Make sure you get your red and your blue pens and your handouts. Oops, don't know why this is all in capitals. Stop it. And when we will go ahead and take our next break, what time is it? It's 9:58. So let's come back at 10:13 and we will restart at 10:13 and I will start the recording at that point. And we'll see how far we can get into the arm today. All right, questions on that? All right, I'll see you guys in 15 minutes. All righty. Let's go ahead and get started. So as I mentioned, we have literally flipped the page and we are on to our next section, which are the muscles that influence and affect the movement of our upper appendage, our upper arm. One of the most obvious and again also misnamed muscles is the biceps brachia. It is not just the biceps. We have almost a half dozen biceps in the body. You're going to be responsible for at least 2 of them. Biceps just means 2-headed, and brachia refers to the region that it is in. So it's the biceps brachia. And this is another one of those muscles that, because it has 2 bellies, it is going to have 2 origins. So here you will see those magic words on the exam, be specific. If we are talking about the origin or the name. They have the same insertion, they have the same actions, but to be able to identify them by name or to identify their origin, you will see those 2 words, be specific, and that's exactly what you need to be. All right. Now, as you notice here, there are 2 heads. Let's go ahead and highlight that. We can see the division between the 2 heads or the 2 bellies. And what are the 2 bellies referred to as?
Short head.
Long head, short head.
Long head and short head. And which is which?
Short head is coracoid process. Oh, wait, hold on. Never mind.
You're right. No, but you're absolutely correct when it comes to the origins. Absolutely. And so you're right. The origin, and we'll go ahead and start with the picture, and then we will go to the bone picture in just a minute. But you're right. The short head is called the short head because it has a short tendon. this short tendon that goes right up to the coracoid process. So notice this one here, the medial one is the one that is the short head. However, the way I actually remember it for me is that means that the L long head is the one that is L lateral. So the lateral is long, and that makes it easy for me to remember. Now, when we look at the models, which we'll do in a second, it can be a little tricky because this short head's tendon is very superficial and easy to see, and it stands out. And I love this picture from your textbook because this picture from your textbook does a great job of showing us the long tendon of the long head. But notice the tendon is very deep. It goes in that notch on the humerus, the intertubercular sulcus. That, remember we said, goes up the neck all the way up to the head. Then notice it actually goes over the head of the humerus into the shoulder cavity, where it goes over the head of the humerus all the way to the scapula, where it attaches to that roughened bump on the top of the scapula, the supraglenoid tubercle. I make a point of emphasizing this because when we look at, where do I have it? Where did I put it? Did I open it here? Must have opened it here. There we go. There we go. This will work. So notice, again, this isn't the best view. Maybe your lab manual has the practice anatomy lab. Let's see if this looks better. None of these do a great job of showing it. All right, so that one does the best one. I can actually bring these up for this. But notice here, what you'll see when you get a chance to look at these things is that the medial short head, you see its entire belly and its entire tendon, whereas the long head actually tucks down and inside and can't actually be seen as easily because it is deep. If we switch views this way. So here, notice the long head hidden by the deltoid. You can't see it, but even if the deltoid was removed, you wouldn't be able to see it very well because of the way that it tucks in. All right, so now let's go ahead and draw them because then that'll bring us to our other point as well. Here we are for the bicep brachia, and again, we need to distinguish our different origins. So what is the origin for the short head again?
Coronoid process.
All right, so we know the short head is
Coracoid.
medial, and it is the coracoid process. So we'll use pink for this to distinguish it. And it's right up here on that bird's beak. All right, whereas we'll use the dark red for the long head, which is lateral. And what is its origin again?
Supraglenoid tubercle.
Excellent.
Glenoid.
Supraglenoid tubercle. So again, it's going to be up here on the top of the glenoid cavity, that little roughened bump up there on the top. And so both of them originate from the scapula. Even though this is a large, prominent muscle sitting over the superficial surface of the humerus, it doesn't actually originate from the humerus. So again, on the exam, if I point an arrow at one of these things, I could ask the question, identify the muscle, be specific, and identify the origin, be specific. And if I say identify the origin, be specific, do not write that the origin of the long head of the bicep brachia is the supraglenoid tubercle, and the origin of the short head of the bicep brachia is the coracoid process of the scapula. That statement is 100% true, but I am only asking you about the one belly. And if I'm only asking you about the one belly and asking you to be specific, you need to be able to tell the difference between the long head and the short head, and just give me the precise piece of information I'm looking for. If I point here and ask you to identify the muscle, don't say bicep brachia. Don't say long head and short head of the bicep brachia. Be specific. And again, I will, if you see those two words, it's the only time you're going to see those two words on the exam is when we're dealing with a muscle that has multiple heads. And so I want a precise origin or a precise name. It's the only time you're going to see it on the exam. So when you see those terms, be specific. Warning bells should go off, but it also should instantly clue you in that we are talking about a muscle that has multiple heads, and you need to be precise about what the head is and potentially what the origin is. All right. Excellent. Now, while they have different origins, they only have one insertion. And what is the common insertion for both of these?
Tendon into radial tuberosity.
Excellent. And again, what we care about is where it attaches to the bone in this case. So that is that radial tuberosity right there. The common tendon is just to remind you that the two bellies come together into a single insertion. So you don't actually have to say that on the exam. You can just say the radial tuberosity. So there you go. And notice, again, this is the one we talked about. You do the curls for the girls. Big, superficial, two-bellied muscles, sits right on top of the humerus. And notice it doesn't even attach to the humerus. It attaches above to the scapula, and below it attaches to the radius. Which tells us something else about this muscle. We know origins are pulled towards, insertions are pulled towards origins. How many joints does this muscle cross?
Two.
Two, shoulder and elbow. Being an anterior muscle, what is its effect on the shoulder going to be?
Flex.
Flex. Being an anterior muscle, what is its effect on the elbow?
Flex.
Right, because anterior muscles flex, right? However, how many actions does this actually have?
3.
3, because the other rule you need to remember is that when we talk about the radius and the ulna, remember we have that special ability to be able to pronate and supinate the forearm. Do both bones move during pronation and supination?
No.
No, which bones move? Which bone moves during pronation and supination?
The radius.
Yeah, the radius moves, the ulna doesn't. What that means is a muscle that attaches to the ulna when the ulna doesn't move, is it going to be able to pronate or supinate the hand? No, but if a muscle attaches to the radius, we do have the ability to pronate and supinate. And which of those two things does the bicep brachia do?
Supinates the radius.
It supinates. So exactly, as that tendon pulls on the radial tuberosity, it twists the hand up into a supinated position. And again, you may not have thought of it in these terms, but you're aware of it. When you're doing those curl exercises, you can do those while supinated. You can also do those while pronated. And can you lift as much weight pronated as you can supinated? No, because when you pronate, you are actually kinking the tendon of the bicep brachia. The bicep brachia pulls the arm out supinated. So when you're pronated, you're actually kinking the tendon and the bicep brachia isn't able to engage. And so as a result of that, you're not able to lift nearly as much weight when you're pronated as you can when you're supinated. So those are our 3 actions. Flex the forearm, supinate the forearm, flex the shoulder. Questions on that? All right, excellent. Now, notice, as I mentioned, when you're pronated, you're not able to really engage your bicep brachia, but you're still able to flex your arm. And the reason for that, or the flex the elbow, you're still able to flex the elbow because the bicep brachia, while the superficial, the most prominent, the one on top, the one that everybody notices, it's not the only muscle that's capable of flexing your elbow. And in fact, if we go back to our illustration from the textbook, again, we see this nice, I'll color it in to make it emphasize it. Bicep brachia that sits here on top, sits on top of the humerus, but doesn't actually connect to it. But if you notice closely, there is an additional muscle immediately beneath it, a deep muscle that sticks out down here on the sides. And this deep muscle that sticks out here on the sides is what is known as the brachialis. The brachialis sits right underneath the bicep brachia. It is deep to the bicep brachia, sits right underneath it. However, it has different attachment points, so it's going to have different functions. Let's take a look at its origins and insertions and see if we can see how that works. What is the origin of the brachialis?
Front distal half of
Yeah, basically it's the front distal half of the diaphysis. In fact, if we remember correctly, here, I'll cheat a little bit and bring this down. If you remember way back in the day when we were learning about bones, there was kind of a raised triangular ridge right here that indicated something. Anybody remember what that bone feature was called?
Deltoid tuberosity.
Deltoid tuberosity, exactly. The deltoid tuberosity is actually basically where the brachialis starts. In fact, we basically have a fancy name for this. When this occurs, we say that the muscles embrace. So basically what happens is our deltoid muscle comes down and basically embraces the brachialis muscle. So they embrace each other. Where one starts, the other ends. It makes it convenient on something like an exam. Let's say, for instance, I had a picture of a humerus on the exam. I could have an arrow that points, or actually this is more something I would do in the classroom because I don't have to be this tricky on the lab exam, I mean on the PowerPoints, but right, this is that deltoid tuberosity. So this is where our deltoid inserts, and this is also where our brachialis originates. You'll see why I'm making a point of emphasizing this in just a minute. But let's go back. Origin indeed was the anterior diaphysis of our humerus. And what's the insertion?
coronoid process of ulna and capsule of elbow joint.
And again, we just care about the, oops, should be blue. There we go. That little bump that sticks out on the ulna. Now, two things. Notice how this muscle sits right underneath the bicep brachia. But unlike the bicep brachia, how many joints does it cross?
One.
Just one, the elbow. And does it insert into the movable bone or into the stable bone?
Stable?
Stable, right? So can it pronate or supinate?
No.
No. In fact, how many actions does the brachialis have?
One.
Just one. All it does is flex the elbow. And does it matter what position the hand is, whether the hand is pronated or supinated?
No.
No. So this is one of the reasons why we're able to flex the elbow even when pronated, because this muscle doesn't care your hand position. All it cares about is flexing the elbow. Again, people often do those pronated curls because it makes their muscles so much bigger. The irony of it is the muscle it makes bigger is actually the brachialis, the deeper muscle. But when the deeper muscle gets bigger, it pushes the bicep brachia out more. So your arms look bigger, even though you're not technically not really working out the bicep brachia. You're just working out that brachialis, pushing it up. Now, last thing about the brachialis. As I mentioned, you are responsible on our bodybuilders for being able to recognize superficial muscles. And the brachialis isn't a superficial muscle. It's a deep muscle. However, as we pointed out, the deltoid comes down and embraces the brachialis. So as it turns out, no matter how big that arm gets, the deltoid muscle is always going to point right at the brachialis muscle. Notice here, this is the long head of the bicep brachia. Here, even though we haven't talked about it yet, is the lateral head of the tricep brachia. But right here in between is that brachialis. So even though it is a deep muscle, because the deltoid always points at it, it is very easy to see on these bodybuilder arms. We can clearly see three defined muscles here on the lateral aspect of their arm. So even though the brachialis is technically a deep muscle, you are responsible for it on the bodybuilders. All right. Questions on that? All right, excellent. There's one, oh, was there a question? Did I talk over someone? No, okay. There's one more major flexor of the arm, and of the elbow. And interestingly, it's actually this muscle down here on the forearm. Normally, we think of forearm muscles as being muscles that affect our hand and our wrist. But on our forearm, we have this very interesting muscle known as the brachioradialis. Here's what I want you to do. If you're sitting at a table or if you're sitting at a desk, I want you to make a fist with your thumb pointing up. Take that fist, put it underneath the table, and as you put it underneath the table with your elbow bent, what I want you to do is push up on the table. And as you push up on the table, you'll notice this muscle sticks out right on top. This muscle that is sticking out right on top is your brachioradialis. Notice, as we can see here from the picture, the brachioradialis lines up right here with the thumb, right on the top, and so you can make that muscle stand out. That is the brachioradialis. Now, like I said, it looks like a forearm muscle where it would affect the hand or the wrist. So what we have to do is look at its origins and its insertions. What is the origin of the brachioradialis?
Lateral supracondylar ridge of humerus.
Excellent. Not the lateral condyle, not the lateral epicondyle, but the supracondylar ridge, that raised ridge on the lateral aspect of the humerus. And what is its insertion?
Styloid process of radius.
There you go, styloid process on the radius. So notice it doesn't cross the wrist. It doesn't cross the hand. In fact, how many joints does it cross?
One.
Just one, just the elbow. Of course, being a mostly anterior muscle, what is the action that it has on the elbow?
Flex forearm. and
Flex.
Yeah, flex the forearm. However, notice it's on the radius. Being on the radius, is that the movable bone or the stable bone when we pronate and supinate?
Movable.
Movable. So that means it should be able to pronate or supinate. And which one does it do?
Both.
Both? How can it do both? Can I have a single muscle that both flexes and extends my elbow? Or flexes and extends my arm? No. So how do I have a muscle that does both here? How can it both pronate and supinate? Well, the answer becomes this. How many of you have waited for a bus before? A couple of you, excellent, all right? For the rest of you, I highly recommend it. When you stood there waiting for a bus, did you stand there in anatomical position with your arm to your side and your hand out with your thumb pointing outwards? Is that how you waited for the bus and it's standing there in anatomical position?
No.
No. Typically, you stand in what is called a body neutral position. Body neutral position is with your palm facing medially and your thumb facing anterior. Remember how we mentioned most of the muscles of your body are at their optimal length for powerful contraction in anatomical position? Well, the brachioradialis is one of the exceptions. Its optimal length, its job is to put your arm into body neutral position so that you can flex that elbow efficiently. So if you're already supinated, it's going to pronate your hand to bring it to body neutral. If you're already pronated, it's going to supinate your hand. So we think of it as two actions where it can both pronate or supinate. But really, if you think about it, it's kind of just one action. Its action is to bring the hand, bring the forearm to body neutral position. Now, we don't have a fancy term for that, so that's why we consider it three. It flexes the elbow, it is going to pronate or supinate the forearm. So flex the forearm, supinate the forearm, and pronate the forearm. And those are its three actions. All right. Questions on that? Excellent. So that is our brachioradialis. Very obvious landmark. And like I said, notice, hopefully you did it. If not, first and foremost, shame on you. But secondly, do it, especially if you're not on camera. Put your hand under that table, make that muscle stand out. And if you think about it, you may not have thought of it in those terms, but it is a very common bodybuilder pose. Bodybuilders are very frequently have this pose where they're pushing down on their hand. And the reason they're pushing down on their hand is to make their brachioradialis stand up. We go back to this guy, notice he's curving it in so that he tries to make it stand up a little bit. And here our bodybuilder, notice our bodybuilder here has her hand twisted. And remember, the brachioradialis lines up with the thumb. So if we follow the thumb over, we follow the thumb straight back, we can see she's trying to make her brachioradialis stand up prominently here as well. Again, it's not going straight up because she has her hand twisted, But if she didn't have her hand twisted, we'd be able to see that. And we can even, although she's not trying to highlight it, see a little of the brachioradialis here, because again, it always lines up with the thumb. That lining up with the thumb of our brachioradialis is going to be huge when we get to the forearm. The forearm is the part that I think people have the most problem with. There are dozens of dozens of muscles in your forearm that affect your wrist and hands. I'm only holding you responsible for 6. They're all superficial. 3 are on the front, 3 are on the back, and that's it. So as far as the forearm goes, you're getting off pretty easy. But even still, it can be challenging. And one of the things that can help, just like driving directions as you're trying to get your way around your city, once you have a landmark that you can use as your base point that helps you to be able to branch out and find what you're looking for from there, it makes it a lot easier. And that's what our brachioradialis is going to be. It's going to be our starting landmark. So from there, we can explore the forearm and hopefully make some sense of it. All right. Questions on that?
I have a question.
Yes.
So on the exam, will we be seeing these bodybuilders and having to identify the muscles on them, or is it just going to be bones?
No. So as I mentioned, yes, the majority of the exam is going to be charts or models of the body, of the parts of the body where you need to identify the muscles and then, know their origins, insertions, and actions. But as I mentioned, probably a third of the questions will be bone pictures. Maybe not a third, maybe 20, 25% will be bone pictures where you'll be getting a picture of a bone and you have to identify the muscle by its attachment point, by its origin and insertion. And as I also mentioned in this test, I haven't finalized this test yet, but this test usually is somewhere in the 75 to 85 questions. I would say of those, probably 5 or 6 of those questions are going to be bodybuilder pictures.
Okay,
And
so
it's not going to be a huge part of the exam, but it may be 5%, something like that. Like I said, 5 or 6 questions. And again, it's not just going to be these bodybuilder pictures. I've got a, you can do a web search and find millions of bodybuilder pictures, but I'm at least using these to give you some examples.
So we will still see like muscle pictures on the exam, not just the bones, right?
Yeah, the majority of the exam will be pictures like this.
Okay.
Where we'll have an arrow pointing to, in this case, a muscle like this. And I can ask one of 4 potential questions. Question 1, identify that muscle.
Brachialis.
Brachialis, excellent. Identify the origin of the muscle.
Front distal half of humerus.
Excellent. Identify the insertion of this muscle.
Coronoid process of ulna and capsule of elbow joint.
Excellent. Identify the action of this muscle.
Flex forearm.
There you go. You could also have questions like this. Again, I will be honest, this will not be the orientation of the picture that I would use for this, but for our purposes right here, I'll use it. Identify the muscle, be specific.
Bicep.
Short head, bicep brachius, brachia.
Is it the short head?
Long head.
Long, remember the long is lateral. If I can see the deltoid, I'm on the lateral aspect of the arm. So it's the long head of the bicep brachia. Identify the origin of the muscle, be specific.
Oh, supraglenoid tubercle.
Of the scapula. Although honestly, if you get supraglenoid tubercle, I'd probably give that to you. Identify the insertion of the muscle.
Common tendon into radial tuberosity.
And again, you just have to say radial tuberosity, but yeah, absolutely, you would get that correct for that. Identify the 3 actions of this muscle.
Flex supinate forearm and flex arm.
Right, exactly. And again, you'll be able to use that to your advantage. If instead, even though it's one we haven't talked about yet, I point here and ask you to identify the 2 actions of this muscle. even if you don't know what muscle this is, you know it's definitely not the bicep brachia. Because how many actions does the bicep brachia have?
3.
You know it's definitely not the brachialis, because how many actions does the brachialis have?
1.
1. And I've asked for 2. So if I've asked for the 2 actions, you know this must be a muscle that has 2 actions to it, and that's going to help you to narrow down what it might be. Anyone happen to know what that happens to be? Identify the muscle, be specific.
Triceps brachia.
Medial
but remember,
triceps brachia.
I had that magical term, be specific. So it is one of the 3 heads of the tricep brachia, but you would have to be specific and identify it's the lateral head. And again, in fairness, we haven't talked about the tricep brachia yet. So it's, in fairness, that's not a fair question to ask. But again, I will be very precise in my wording. When you see the terms be specific, you know I'm asking for a multi-headed or muscle. If I'm asking for 1 action or 2 actions or 3 actions or 4 actions, you know exactly how many actions that muscle must have, and that should be able to help you to narrow it down. So again, if I ask for 2 actions, you know it's not the brachialis. You know it's not the bicep brachia. You know it's not the deltoid. There's a lot of muscles you'll be able to eliminate right away because you know they don't have 2 actions. They have 1, or they have 3, or they have 4. So yes, this will be the majority of the test. But like I said, about a third of them would be something like this, where I have a picture like this here, and I have an arrow like this, and I say identify the muscle that inserts, pardon me, the muscle that originates from this bone feature. And what would your answer to that be?
Brachialis.
Brachioradialis.
Brachioradialis, right? That's the lateral supracondylar ridge. Excellent. So we have that. And then, like I said, not more than a handful, 4, 5, 6, something like that, but it's not going to be more than that, will be pictures like this, where I say, identify the muscle.
Trapezius.
Yeah, there you go. Add in the histology, and that's pretty much this next exam.
So for histology, are we going to be looking at histology for the smooth, the skeletal, and cardiac muscle?
Yes, which remember we did on the very first day of this section. We looked at all 3 different types of tissue under the microscope. We looked at cardiac muscle, skeletal muscle, smooth muscle, saw how to identify it under the microscope, and then talked about their characteristics. We will do a little bit more histology on the last day, but you also have your handout to remind you. We also have those models. Remember we saw in the, where is it?
The skeletal anatomy, is that what you're talking about?
Cell. So when we go back here, go back to your modules. So not only are you responsible for histology, but remember we had some histology models here as well. So where were those pictures? Muscle classroom models. So remember, these were the tissue types that we needed to know the characteristics: skeletal muscle, smooth muscle, cardiac muscle. So I could show you them under a microscope, or I could show it here, identify the organelle, identify the structure, yammer. But remember this is a model of a skeletal muscle cell that shows the proteins, the myofibrils, the triad, the transverse tubules. This one, remember, even had a satellite cell on it, the endomysium. We have it from a couple of views. There was the second one also showing a muscle cell, transverse tubules, great sarcoplasmic reticulum, great triad, right? We see the myofibrils, the dark and the light stripes, all of those. And then we even had that sarcomere model where we could actually see the proteins, the myosin, the myomysin, the Z-disc, the actin. We can even see the yellow stripe, which represents the tropomyosin, and the little green knobs that represent the troponin. So our histology, we also have these great models for as well. So these are, and of course, microscope slides. And I think that's it. I think that's pretty much what's going to be on this exam. again, none of these individual pieces of information are hard by themselves. The challenge of all of this is just so much of it, and you have so little time. But again, the goal of the lab exam is to be very straightforward. These are the things that, I have no problem telling you exactly the types of things that are going to be on the exam, because I know you're going to have to work hard to learn them. So yeah, I can't think of anything else that would be on the exam. Histology slides and models, bones, bodybuilder pictures, but mostly it's going to be pictures of models or charts or things like that that show the muscles. And again, you've got some great resources to study those from. Not as great as being in the classroom obviously would be, but as we talked about, you have your practice anatomy lab, which does a great job of showing, notice here again, that deltoid points right to the brachialis, right? There's that long head of the bicep brachia that we see here, and we haven't talked about the tricep brachia yet, but we have that there. Remember your virtual anatomy lab from Cosumnes River has got muscle pictures of the torso. So again, we've got the torso, the limbs, we're on the limb now. So here's some arm pictures. And again, the nice thing about it is it shows the pictures so that you can figure it out by yourself. And then the next slide shows you some of the labels.
So is the practice anatomy lab, that activity, is that on My Mastering or?
Yeah, so again, yes, the practice anatomy lab is the one that's here in your My Lab and Mastering, practice anatomy lab. And then notice the first part is the cadaver, but if you go into the anatomical models, it shows that here. And even though I showed it to you, I'll show it to you again because we literally just did it, so it's a good refresher. Notice also, so again, here in the models, we can go to the muscular system. We can see, for instance, the upper limb and stuff like that we were looking at. However, notice if you go back to the home page up here under animations, upper limb, we just did the bicep brachia. Remember, I showed this to you on the first day, but again, we've covered a lot of material since then.
Biceps brachii is the large two-headed flexor muscle located in the anterior compartment of the arm. The origin of the long head is from the supraglenoid tubercle of the scapula. The origin of the short head is from the tip of the coracoid process of the scapula. The two heads insert by a common tendon onto the radial tuberosity and by the bicipital aponeurosis, which blends with the deep fascia on the medial aspect of the forearm.
And again, remember, you just need radial tuberosity.
Biceps brachii flexes the forearm at the elbow supinates the forearm, and weakly flexes the shoulder. It is innervated by the muscular cutaneous nerve.
So notice, just like we're drawing it, this actually draws the origins insertions for you, and these animations actually show you the actions. So it's a great way of studying this. And remember also, as we talked about under here, is it here? No. If we go back to the study area, I don't know how to get back to the study area from here, other than to go back here, My Lab and Mastering. There may be a way to get through it through the practice anatomy lab. I haven't messed with it enough, but there's actually a flashcard maker. So you got a flashcard maker where you could do the anatomical models. You could do muscles of the upper limb, or pick all the, and then you can actually pick the muscles you wanted to identify. And then when you go and either take a quiz or a review on them, it shows you the muscle and you have to identify what it is, right? And then you can flip the card. There's a flip the card to see what the answer is, right? And again, notice it's not giving you long head and short head, so it's not as ideal that way, but it's still not a horrible tool. And these can actually be downloaded to your smart devices. So your cell phone or your iPad, so you can be studying these flashcards can actually be made to download to your device and be able to do it that way. So there's some great resources. Yeah, here it is right here. See, there you go, mobile flashcards. So you click up here to the mobile flashcards and you can make them for your smartphone or for your tablet. So there's some great, great tools that if you're not taking advantage of, first and foremost, shame on you, but more importantly, take advantage of them, especially if you're paying for them. So by all means, do these things so that you can be successful. All right. Excellent. We got a little sidetracked there, but I think it's definitely worth it because again, we're putting a tremendous amount of time and effort. You and I both are putting a tremendous amount of time and effort into this class. It's important to do what you can to be successful, right? Last thing we want to do is put all the time and effort into this class, not be successful and have to do it again. I have to do it again, but at least I get paid. You shouldn't have to do it again. So do the best that you can to be successful with this. And hopefully these are things that can help you to do that. All right. All right. Like I said, I'll get off my soapbox. I'll put my pom-poms away. Questions on any of that? All right. So we did our 3 big flexors of the elbow: the bicep brachia, the brachioradialis, the brachialis, and the brachioradialis. Those 3 muscles there. Excellent. Let's move on to another important muscle of the body. This is a very lateral muscle, and not being surprising, notice it's not anterior, it's not posterior, it's mostly lateral. So what kind of an effect is a lateral muscle most likely to have?
Abduction.
Yeah, abduct the arm, bring that arm up. Now, notice we already mentioned how the deltoid embraces the brachialis, but the deltoid actually embraces not 1, but 2 other muscles. Let's go back to our origins and insertion handout. And look at the deltoid. What is the origin of the deltoid?
Acromial end of clavicle, spine and acromion of scapula.
There you go. So the spine, oh, yeah, make this bigger. The spine of the scapula, the acromion of the scapula, and again, the lateral third of the clavicle. Why does that sound so familiar to me?
Same as the trapezius.
Yeah, remember, this is the exact same insertion of the trapezius. So basically, the trapezius comes in and inserts on these bone features where it embraces the deltoid. The deltoid then, as we kind of already indicated at, inserts into the deltoid tuberosity, that triangular region right here on the arm, which, as we mentioned, is exactly where our brachialis begins, on the anterior side. So we got that brachialis on the anterior side starting there. And so that embraces, and again, in the definition, when I'm asking for origins and insertions, you don't have to use the term embraces in there. That's really there just to remind you of how these things kind of fit together. So the trapezius embraces the deltoid, which embraces the brachialis. All right. Questions about its origin and its insertion? So notice one more thing about the deltoid. While it is a lateral muscle on the lateral part of the body, so clearly it is going to abduct the arm. Now that we've seen the origin, some of its fascicles are on the front of the arm and some of the fascicles are on the back of the arm. So what 2 other actions does this muscle have?
Rotate the arm.
Yeah, and again, remember when we talk about a structure that is not on the midline, we think of it in terms of how the anterior surface moves. Notice if my anterior muscle fascials contract, it would pull the anterior part of my humerus towards the midline, which would be a medial rotation. Whereas if the posterior ones contract, it would be a lateral rotation, pulling the front of the humerus away from the midline. So this can both medially and laterally rotate the humerus. This one's a little tricky. Has anyone come up with a good activity that would help us to remember the actions of the deltoid? No Davisites on here, or at least anyone willing to admit to worshiping at the altar of the frog tunnel?
Kelsey.
Ah, Emily. All right, so Emily knows. I did both my graduate and undergraduate work at UC Davis. So I spent more time in Davis than any one living human being should be allowed. And as Emily can tell you, because she lives in Davis, you are actually required by law to spend at least an hour and a half on your bike every single day. Required by law. You'll get a ticket if you don't do it. So when you're out on your bike, you of course need to be able to signal where you're going. And so how do you do that? You put your arm out, you bend your elbow, and if you're going to stop with your arm abducted, you would medially rotate your arm, and that would tell everybody that you were stopping. If you were getting ready to turn right, you would abduct your arm and you would laterally rotate your humerus, and you would tell everybody you're going left. So that ability to signal while your arm is abducted is because you're able to medially and laterally rotate the humerus. And so given all those bike signs when you're in Davis is a perfect function. It's why all Davisites have massive, massive deltoids. Actually, just the one, just the left side from all that time they spend. That's how you can tell. All their shirts sit unevenly because of that massive, massive left deltoid from riding their bikes all the time. All right, excellent. See, and Emily, yep, you saw it with Emily. Yep, she's got it there. There you go, exactly. So there you go. Again, I think hopefully that will help you to understand what we're talking about with those rotations. I know the rotations can be a little tricky, but I think that that hopefully will help with that. All right, questions on that? All right, excellent. That is the deltoid. Another big mover of the upper arm is latissimus dorsi. Now, remember earlier, we were talking about that trapezius muscle that is and if you remember when we talked about the trapezius, it was a superficial, superficial, superior, mostly posterior muscle. Because it snuck a little bit onto the front. And the same thing is true for our latissimus dorsi. Our latissimus dorsi is also superficial. It's a big one on the top. It's inferior. It is below, not deep to, but below the trapezoid. And again, it is mostly posterior. Once again, I think we see this best when we actually draw the origins and the insertions. So let's do that for our latissimus dorsi. Let's start, I was going to say easy, but I'm not sure it's easy. Let's start abundantly with the origin of the latissimus dorsi. What is the origin of the latissimus dorsi?
There's a lot.
Yes, there is. So let's go through it bit by bit. Give me some of it.
Thoracolumbar fascia.
Okay, excellent. So there is that. Remember, we talked about that connective tissue fascia. Of course, we can't draw that very easily, but we know it's there. Maybe we'll draw it to the side as a reminder that it's there. That's thoracolumbar fascia. What else?
Spines of lower 6 thoracic vertebrae.
Excellent. So again, we can count our way up. Here's 12, 11, 10, 9, 8, and 7. So basically from there to there. What else?
Lumbar vertebrae.
Spinous process of all of our lumbar vertebrae. What else?
Lower 3 and 4 ribs.
Some of it's over here on the ribs. What else?
Crests of sacrum and ilium.
Excellent. So the crest of our sacrum, the crest of our ilium, and
Inferior angle of scapula.
there you go. This is a big, huge, broad muscle with a massive number of fascicles, massively convergent. Some of them are coming from the smaller back. Oh, wait, hold on, let's do this. Make the lines very small, make them black. Some of them are coming from the hip bone. Many of them are coming from the spine. A couple are actually attached to the ribs. These are coming off, and some are even starting here, coming off the inferior angle of the scapula. But notice all of these fascicles, all of these fibers from all these different locations are all coming together into a single small insertion. And what is the insertion of our latissimus dorsi?
Intertubercle sulcus of humerus.
Yeah, the intertubercular sulcus. Which, if I remember correctly, is on which side of the bone? Is it? Can we see it on this picture? Or can we see it over here on the pectoralis major picture?
Anterior.
Yeah, notice it's on the anterior side. It is actually located, and I'll cheat and draw it first here in green. Remember, it is here in between the tubercles on the anterior side of the humerus. So technically, I don't see it on this one. So what I usually like to do is I draw a dotted line here to remind myself that I'm not actually looking at it. It's on the anterior side. I may even write anterior here to remind me while I'm doing that. But all these fascicles are going to wrap up around, no, don't want that to be below, to the front of the humerus where they're going to attach on the anterior side. All right, now that we've drawn that, let's look at a couple pictures. Notice as we see how good I am. Oh, so good. Excellent. Notice as we see here, again, big, huge, massive origin. Thoracic vertebrae spine, right? T7 all the way down to the sacrum. Some of them starting up here on the iliac crest. We see that big, huge thoracolumbar fascia that is supporting some of these tendons. And we can even see a little bit that might be connected down here to the inferior angle of our scapula. And all these fascicles, notice, are coming in, and we're losing it because it's actually going to the front of the arm. Where we actually see this best is on the chart in the classroom. Notice here on the chart in the classroom with the arm up, we can actually see, that brown doesn't show up. We can see how that latissimus dorsi is coming off of the front, off of the back, and attaching to the front of the humerus. So basically, this is a big, broad muscle that forms this strap that comes up and attaches to the anterior side of the arm. It's an interesting position, but the nice thing about it is the location of the muscle tells us its actions. Now, technically, it only crosses one joint. And that one joint it crosses is the shoulder joint. But as we know, the shoulder joint is the most flexible of all the joints, the most dexterous of all the joints. So let's go through the list. This is a posterior muscle. Being a posterior muscle, what effect is it going to have on the arm?
Extension.
Yeah. Notice also its origin is on the midline. So it is a medial muscle. And since the deltoid, which was lateral, abducted, what will a medial muscle do?
Adduct.
A deduct. Excellent. But there's a third action. Notice because it comes up under the arm to the front of the humerus. When it contracts and is pulling the arm inward, it also can twist the humerus. And when it twists the humerus, the front of the humerus goes towards the midline. So what type of rotation would that be?
Medial.
Exactly. So because of its anterior insertion, it gives it a medial rotation. And so those are the three actions: extends, adducts, and medially rotates. And again, you may not have thought of it in these terms, but what are the type of people? What type of people have really pronounced latissimus dorsi muscles? Again, I'll give you a hint. The Olympics are right around the corner.
Swimmers.
Swimmers.
Swimmers, absolutely, right? Heck, what's his face?
Michael Phelps.
Yeah, Phelps practically has bat wings, right? Because again, if you think about their action, this is bringing the arm in, this is bringing the arm back, extending it, and medially rotating it, right? If you alternate that with your arms, you're doing the freestyle. If you do them both at the same time, you're doing the butterfly, right? So Phelps has massively pronounced latissimus dorsi, and from what I'm told, he has a very pronounced sternocleidomastoid as well. All right. Questions on that? All right, excellent. So that is the latissimus dorsi. We make a point of emphasizing these aspects because notice while that was a mostly posterior muscle that inserts into the intertubercular sulcus, it's not the only muscle that inserts into the intertubercular sulcus. The other muscle that inserts into the intertubercular sulcus is the pectoralis major. Let's look at the bones for this one. Again, a big convergent muscle. I've cheated and already told you that the insertion is going to be that intertubercular sulcus right next to the latissimus dorsi. But this is an anterior muscle, so its origin is very different. What is the origin of the pectoralis major?
Clavicle. Sternum. Cartilage of ribs 1 through 6.
There you go. So clavicle, sternum, a little bit of the cartilage from root 1 to 6. Anything else?
Aponeurosis of external oblique muscle.
Yeah, remember that aponeurosis, it attaches to the aponeurosis as well. Embeds in it. Your book's got a great picture that shows this. I'll show it to you in a second, but if I leave this page, we'll lose our drawing. Notice our pectoralis major has this very landlocked type of origin. And again, you maybe not have thought about it in those terms, but when our bodybuilder is building up that pectoralis major, can that muscle really spread superiorly or inferiorly or even medially as a result of that? No. So typically when they work out their pectoralis major, it really sticks out and becomes prominent because that's really the only direction it can expand. Right? So again, notice if we go back to our picture here, They've done a nice job of pulling it up a little bit underneath where we see it attaching to the aponeurosis of the external oblique, attaching to the sternum and the cartilage of the ribs and the clavicle up here. And then going into that same insertion as the latissimus dorsi. And let's go back here. Because remember, with our latissimus dorsi, as we mentioned, this is a posterior muscle. And so because of that, it affects the shoulder and it extends the arm. It is a medial muscle, so it adducts. And because it attaches to the anterior insertion, it gives us a medial rotation. Right? Those are kind of how the location of the latissimus dorsi affects its movements. Well, let's look at the pectoralis major. The pectoralis major is an anterior muscle. So what effect is it going to have on the shoulder?
Flexing it.
Flex.
Flex the arm, flex the shoulder. Oops. Excellent. It is also a medial muscle. So what effect is that going to cause?
Rotating it.
True, it's going to rotate, but also because it's medial and pulling towards the midline, it's also going to adduct. But you're right, because of that anterior attachment, that anterior insertion, it is also going to medially rotate. So notice both muscles adduct the arms, both muscles medially rotate the arms, but the pectoralis major is on the front, so it brings the arm forward. The latissimus dorsi is on the back, so it brings the arm back. Two very different muscles in very different locations, but because they have the same insertion point, they are very similar in their function. Excellent. Questions on that? All right. One last major muscle of the upper arm that we are responsible for the origins and the insertions for, and that is the tricep brachia. So the good news is this is the last one we need to know origins and insertions for. What is the bad news?
There's 3 of them. Got to be
Yeah.
specific.
It's a triceps brachia. So there are 3 heads that we need to be specific about and 3 origins that we need to be specific about. So let's switch to our bones to help us with that. The tricep brachia is the prominent posterior muscle of the upper arm. And what's nice, and I will cheat a little bit. Actually, let's go back to the picture here for a second and look at it. What's nice about this is that it has one big major tendon that all 3 bellies connect to. It's very prominent, easy to see on the posterior, connects to the bump of the elbow. And from that, we can find the 3 muscles because all 3 muscles are going to be associated with it. Notice over here, lonely and by itself, we have the lonely lateral head. So our lateral head is lonely, all over here by itself. The smallest head is the medial head. So our medial head is mini. And of course, our third head is the long head. Change colors. And the long head happens to be so long that it goes all the way past the humerus, all the way to the scapula. So of course that means that our long head is long. So there you go. Our lateral head is lonely, our medial head is mini, and our long head is long. And notice all three of them attached to the same tendon. So if you find that tendon, you can find the three muscles. Notice if we go back to one of our bone pictures, see what we've been looking at here. I've gotten rid of everything. Right, here we go, perfect. Let's get back away from the pictures. Here again is that tendon. With that tendon, notice I can clearly see, especially since this is the deltoid, this is my lonely lateral head. Even though that pesky deltoid's in the way and I can't really see where it's going, I can see that this is my long head on its way up to the scapula. And this view, I really can't fully see the mini head, but we can see just the teeniest bit of it right here where it's getting ready to turn the corner. That mini little medial head, which again, since we're looking at the lateral aspect of the arm, it's not surprisingly we can't see the medial head on the lateral part of the arm. That seems pretty reasonable. But if we turned it over, right, now we're looking more at the medial aspect. We can see a little bit of that medial head right there sneaking its way onto the medial aspect. And of course, the long head going up to the scapula. And again, notice, not surprisingly, from a medial view of the arm, we really can't see the lateral head. Again, hopefully that's pretty intuitive. All right. So we have the pretty picture from the textbook. We have some models and some charts that show it nicely. Let's draw the origins and the insertions. So let's start first with the medial head. Now, the medial head, obviously, not surprising, more than anything else, is going to be medial. That makes it easy. And it's part of the tricep brachia, so it is going to be posterior. But it's also small. If it's small, does it get to the scapula? In fact, does it get anywhere near the scapula?
No.
No. So it's also distal. So when we look at the origin of the medial head, the origin of the medial head is essentially the posterior, distal, and medial aspect of the humerus. When we look at the lateral head, not surprisingly, it is going to be lateral. It is going to be posterior, because after all, it's the tricep brachia. And this lonely one gets to go a little farther up the arm. So this one is more proximal. So when we look at the origin of the lateral head, basically it is the posterior lateral proximal surface of the humerus. And that brings us to our long head. Our long head, of course, is long, so long that it doesn't stay on the humerus. And instead, where does it go? What's the origin of the long head?
Infraglenoid tubercle.
Excellent. Infraglenoid tubercle. which of course we see very nicely here, is that roughened bump underneath the glenoid cavity. Notice, and I'll draw it in green here so that we don't get confused, this also does a good job of showing us the supraglenoid tubercle. So there you go. Those are our 3 origins for our tricep brachia. Questions on that? And let's go ahead and get rid of that so it doesn't confuse us. Excellent. 3 different names, 3 different origins, but how many insertions?
1.
Just 1. What?
Olecranon process of the ulna.
There you go. The bump of the elbow. All right. The olecranon process of the ulna, the bump of the elbow, that's what it inserts into. So we've done the origins and insertions. Now we can do the actions. How many joints does this cross?
2.
2. Thanks to the long head, it crosses the shoulder and it also crosses the elbow. But it's on the ulna. Is that the stable bone or the movable bone?
Stable.
Stable bone. So how many actions does the tricep brachia have?
2.
2. Extend the elbow, extend the arm. There you go. 2 actions. All right. Questions on that? Excellent. Just that easily, we have identified all the muscles that we need to know the origins and the insertions of for the arm. Now notice there's still a lot of muscles left on this list. And I want to talk about the first few of these first. But to do that, I want to come back to, hold on. What I want to do is come here. Excellent. Again, to help us with some of this information, I absolutely positively guarantee you there is going to be a picture of the scapula on the exam, because if you think about it, there is a ton of stuff that we have talked about with the scapula. There are arrows galore on this, all right? Again, we've already done some of them, so let's do some review. This picture, this arrow, identify the muscle that inserts into this bone feature.
Trapezius.
Pop quiz. We can either do this here or I can make this a quiz and you guys can take it right now. Which would you like to do?
Trapezius.
Excellent. Same bone, same arrow. Identify the muscle that originates from this bone feature.
Deltoid.
Excellent. And notice I could have easily done the same thing by having my arrow out here on the acromion. Let's do another one. Identify the muscle that inserts into this bone feature.
Brachialis.
Nope, although that's a good guess. Identify the muscle that inserts into this bone feature.
Pectoralis minor.
Pectoralis minor. Identify the muscle that originates from this bone feature. Be specific.
Short head of the biceps brachia.
Excellent. Identify the muscle that originates from the bone feature. Be specific.
Long head of the biceps brachia.
Excellent. Identify the muscle that originates from this bone feature. Be specific.
Long head of the triceps brachia.
Excellent. Let's go back just for the heck of it. Identify the muscle that originates from the bone feature.
Latissimus dorsi.
Yep. Ooh, and that just reminded me of two more. Identify the muscle that inserts into the bone feature.
Rhomboids.
Nope. Close though. Notice this is the anterior surface, no spine. So what inserts into the anterior medial border?
Serratus anterior.
Serratus anterior.
Here we have the spine. So what would be the muscle that inserts into this bone feature?
Latissimus dorsi.
Inserts in this bone feature. You said it already.
Rhomboid.
Rhomboids. Right?
Rhomboid.
Rhomboids, remember, inserts into the medial border of the scapula, and the serratus anterior inserts into the the anterior surface of the medial border. So posterior medial border, rhomboids, anterior medial border, serratus anterior. Excellent. Now, more fun. Someone remind me again what this indentation above the spine on the scapula is.
Supraspinous fossa.
Excellent. Fossa is an indentation. This indentation is above the spine. So this is the supraspinous fossa. Now, while you don't have to know the precise origin and insertion, there happens to be a muscle that sits in this space. What muscle do you think sits in the supraspinous fossa? Now, if only I had a list of the
Trapezius.
muscles. If only I had a list of the muscles I was responsible in front of me, I might be able to figure out what muscle that I'm not responsible for the origin and insertion of. There you go, Samuel's got it. What sits in the supraspinous fossa? The supraspinatus. If I remember correctly, there's an indentation below the spine. What was that called?
Subscapular fossa.
Now, beneath the spine was what?
Sorry,
Infra.
fossa.
Infraspinous.
Infraspinous fossa. And guess what muscle sits in the infraspinous fossa?
Infraspinatus.
Infraspinatus. Excellent. What was the indentation on the front of the scapula where there's no spine?
Subscapular fossa.
And what muscle sits in the subscapular fossa?
Subscapularis.
Subscapularis. Excellent. Notice, ooh, do I have this? Let's see how clever I actually am. Where do I have this? I wonder if they have it. No. What did I do with that? Yeah. Apparently, I'm not nearly as clever as I think I am. Shoot. Alrighty. Yep. Okay. there is a great model in the classroom, which apparently I don't have a picture of, although I would have bet money I had it at some point. I will get it and provide it to you. That actually shows the muscles in these locations. So we have, again, this scapula, and not surprisingly, as we just finished talking about, we have the supraspinatus, which basically sits in the supraspinous fossa, the infraspinatus, which sits here underneath the spine, the subscapularis, which sits here in the subscapular fossa. And if we go back to our model, notice here on the anterior side, we see this here would be the subscapularis on the anterior part. And hopefully, no, all right. They show a little bit of it. It's better when the deltoid is taken off. Let's see what our practice anatomy lab shows us. Nope, not the cadaver. Nope, that doesn't help us either. All right, tack with it. Let's go to the pictures in your class, in your textbook. All righty. So here we have these 4 sets of muscles.
Not sure if you can see it very well, but
Oh, that works. That's pretty good. Yeah, absolutely. So that's a good example of that. What's that on? Is that in one of your? Where's that picture?
it's an app that I found that
Cool.
used for study.
So notice if we look like you're doing at the posterior side, here we see it here. So this is what you're looking at. This is what you're showing us here. Notice we can see the supraspinatus, which is up here in the supraspinous fossa. The infraspinatus, which is this muscle right here. And notice there's one more really big, obvious, prominent muscle as well. This muscle that actually sticks out from the side and actually has a little bit of a gap, a little bit separate from the rest of the shoulder. Any idea what this muscle I just highlighted here is? And actually, let me change colors of that. What is this muscle here in green? Any idea?
Teres minor.
Well, not a bad guess, right? Obviously, it's the next one on the list, so that should be what I'm pointing out to you. But if you were like me, I would have just cheated and followed the lead out and seen it was the teres major. Now, is the teres major one of the muscles you're actually responsible for?
No.
No. However, what it is directly across the gap from our teres minor. So here is the teres minor. The teres minor is basically nestled underneath the spine of the scapula, right next to the infraspinatus. So while the teres minor is not a muscle you're responsible for on your list, you can easily use it as a landmark to find the teres minor, which is right across from it. All right. Notice one other thing about these 4 muscles. So the supraspinatus up here, infraspinatus right here, teres minor right here, and on the front side, the supraspinatus, which is this big, huge muscle. Why are you not? There you go. There it is. That attaches here like this. These 4 muscles are the only muscles whose tendons actually come together and surround the head of the humerus. So these are the only muscles whose tendons help to come around and support the head of the humerus. It's almost like they wrap around them providing this really important support structure for the head of the humerus, locking it in the place, almost like a cuff-like structure that would stabilize the head of the humerus while it was rotating around in space. And what would a good name for this muscle group that provided the support for the head of the humerus be? Let me say that again. It's almost like a cuff-like structure that supports the head of the humerus while it rotates in space. Come on, I'm not giving you this one.
The reticulum.
It is a
Reticulum.
cuff-like structure that supports the humerus while it rotates in space. It is a cuff-like structure that supports the head of the humerus while it rotates in space. While the humerus rotates in space, there is a cuff that supports it.
Rotator cuff. Sorry.
There you go. See how easy that was? Yes, so it's the rotator cuff. There you go. All right. If you are a pitcher or know a pitcher, if you know a quarterback or are a quarterback, right? You hear plenty of times where pitchers and quarterbacks and people like that who put a lot of strain on their shoulder have a rotator cuff injury. Well, typically that is damage to the tendons or one of these four muscles, because these four muscles form that supportive cuff around the head of the humerus, anchoring it and supporting it in place. So another muscle group you are responsible for, that we also need to know the individual muscles. Now, we don't need to know the origins and insertions, but their locations help us to understand their functions. Let's start simple. The supraspinatus basically comes right over the top and straight down onto the head of the humerus. Right? So when it contracts, what effect is it going to have on the arm?
Abduct.
Abduction, exactly. And notice, because it's right on top, there's no rotation, there's no flexion, there's no extension. How many actions does the supraspinatus have?
One.
One. All it does is abducts the arm. And really, only about the first 30 degrees. After that, it's the deltoid that brings it up the rest of the way. So the supraspinatus basically lets you do the chicken dance, but isn't much good for anything outside of that. Let's switch pictures back to here and notice that while we have this big broad spinatus, basically it comes straight across to the head of the humerus. So pretty much, again, in providing that support, it just basically pulls the anterior surface of the humerus towards the midline. So what is the one and only one action of the supraspinatus? Pardon me, not supraspinatus. Sorry, the subscapularis. Sorry, subscapularis.
Medial rotation.
There it is. Hold on. There we go. Yeah, medial rotation. So the subscapularis, medial rotation. So supraspinatus, one action. Subscapularis, one action. But then when we get to the back, notice because of the spine, the infraspinatus is a little bit more inferior. So it is a little bit more angled. Again, we want to provide support for the humerus. So not surprisingly, to provide that support, it is going to bring the humerus back and laterally rotate it. But because it's a little bit inferior, it gets a little bit more of an angle to it. So what else can it do?
Adduct.
Adduct. So again, it is bringing it in and laterally rotating it. right? So like, again, when you're doing those jazz hands, abduct, lateral rotate, jazz hands, right? As a result of that. And even though it's the smallest of the muscles, notice the teres minor has the biggest angle to the head of the humerus. So while it's the smallest of the four muscles, it's the one that has three actions. Now, not surprisingly, it is directly under the infraspinatus. So it also is going to adduct. It also is going to laterally rotate. But because it's even more inferior, it gets more of an angle. And what else can it do?
Extend.
Extend, bring it back. So it brings the elbow in, right? Laterally rotates, adducts. So this is the tennis one. When you're doing that forehand, right? You want to keep your elbow in for that forehand. Otherwise, you get that stress on the elbow, get that tennis elbow as a result of it. So this is that forehand tennis action with the teres minor. So there you go. Four muscles, all wrapping around the head, all associated with the scapula. And for the next class, let me write myself a note about this. I will get that picture of I've got it somewhere because I know I've done this before. Is that scapula model, because it does a great job of showing these muscles and showing that rotator cuff and doing a really, really nice job. So I've got that picture somewhere, but rather than continuing to scramble and try to find it, we'll save it for next time. All right. Questions on that? All right, excellent. All right. We've covered a lot, and I know you're all tired. So here is what I'm going to do. I don't want to start on the forearm because, like I said, the forearming is tough and challenging. But I want to strongly encourage you this weekend to start working on it. I'm going to go through this once quickly to emphasize to you how to study this. And then I want you to work it on your own. We will go over this together. This will be where we'll start. Actually, we'll start with the scapula model, but we'll go straight from the scapula model into the forearm, and that is where we will start on Monday. But remember, Monday is basically 3 days before the exam. So we don't want to wait till Monday for this to be the first time that you're thinking about this. So, as I mentioned, to master the forearm, there are two things you need to do. The first thing you need to do is you need a good starting point. And the second thing you want to do is follow the tendons. The tendons tell you everything about the arm. Lastly, you want to remember anatomical position. Because after all, if you have anterior muscles, what do anterior muscles do?
Flex,
What do posterior muscles do?
stand,
Right? When we have lateral muscles, what do lateral muscles do?
abduct,
And what do medial muscles do?
adduct,
And we can add one more thing to that as well. In the forearm, we have two parallel bones. What is the lateral bone of the forearm?
radius,
And what is the medial bone of the forearm? There you go. So again, I'm going to do this super quick. We will go through this again. We'll look at the models. We'll look at the charts. We'll go through all of this, but I want to do it right here first, just real quickly so you can see using these rules, you can easily pick this out. You need a starting point, that popping up brachioradialis muscle. So here is that brachioradialis muscle. It's that nice, obvious muscle that sticks out on the side of the thumb, is big and prominent. And right next to it is a muscle that is very small and has a very long tendon that goes to the backside of the hand on the thumb. Being a posterior muscle, it is an extensor. It crosses the wrist or the carpal bones. It is on the thumb side, so it is a radialis. And it has a very small belly and a very long tendon. So it's the extensor carpi radialis longus. Big mouthful name, but it tells you everything about it. Tells you where it goes, and it tells you what it does. Being an extensor on the posterior side, what is it going to do?
Extend the forearm.
Extend the wrist.
And
All right, it's an extensor of the carpals. It extends the wrist. Being a radialis, and on the radial side, that means it's a lateral muscle. So what else is it going to do? Right, is it going to abduct or adduct?
Abduct.
Abduct. There you go. The name tells you everything about it. And here's one other thing the name tells you. When you have a longus, guess what else you have?
short.
A shortest. Now, they don't use shortest. They use the term brevis. But yeah, if you have a magnus or maximus, you have a minimus. If you have a longus, you have a brevis. And so not surprisingly, right next to the extensor carpi radialis longus is an extensor carpi radialis brevis. But do you need to know that?
No.
No, so you don't care. But we need to know it's there so we know to hop over it. Because when we hop over it, we find a muscle that when we look at this muscle up here in the top, oops, doesn't look like much. But remember, what are we supposed to do? Follow the tendons. And if we follow this muscle down, what we notice is this muscle tendon branches to go to 3 of my fingers. So what do you think we call this muscle with the branching tendons that go to 3 of my fingers?
Extensor digitorum.
Extensor digitorum, exactly. And guess what the extensor digitorum does?
Extends the digits.
There you go. Notice not all of them. The thumb's got a dozen plus muscles that move the thumb. We're not going to even worry about the thumb because it opposes and does all those things like that. But notice it also doesn't go to the pinky. Pinky is very important if you're Dr. Evil, right? $1 million. And if you're Dr. Evil, you also need a mini-me. So to go to the pinky, we have an extensor digitorum mini-me or minimus. And again, do we care?
Nope.
Nope. But we know we have to hop over a muscle again. And when we hop over that muscle, we find a muscle whose tendon goes to the backside of the pinky. And guess what we call the muscle that goes to the backside of the pinky?
Extensor carpi ulnus. Ulnars.
There you go. Extensor carpi ulnaris. And just that easily, from our starting point of the brachioradialis, we have identified the 3 muscles we are responsible for on our posterior part of the arm. Now, I do want you to notice one other thing while we're here on the posterior side of the arm. Notice while we're over here on the posterior side of the arm, we see the majority of a muscle belly here. Since we're seeing the muscle belly on this side, it must be a posterior muscle, right? Well, not so fast. We identify muscles by where their tendons go. And it turns out this tendon actually goes to the front of the hand. It's on the pinky side, right next to the extensor carpi ulnaris, but this one's tendon, unlike the extensor carpi ulnaris, whose tendon comes to the back of the hand. Oops, no. Whose tendon comes to the back of the hand. This muscle right next to it, its tendon goes to the front of the hand. So guess what we call that muscle that goes to the front of the hand?
Flexor carpi ulnaris.
There you go. It is the flexor carpi ulnaris. Notice if we flip the arm around to the anterior view, notice we don't see nearly as much of the belly of the flexor carpi ulnaris. Its belly is on the back of the arm, but we don't care where its belly is. We care where its tendon goes. And lo and behold, here's its tendon coming to the front of the hand on the pinky side. So that is the flexor carpi ulnaris. It's a prominent tendon. In fact, and I can actually do this better in my right arm, so I'll take off my watch for a second. If you contract and tense, you can actually see not one, not two, but three prominent tendons that stick out in your forearm. Here on the pinky side, that is that flexor carpi ulnaris. But notice there's two more very prominent tendons, three prominent tendons that you can make stand out. And lo and behold, as I look at my arm here, oh, I got to reset my watch. Got to make sure I get my movement points for moving my arms around in class. If we look, lo and behold, here is a prominent tendon. And here is a prominent tendon. So let's look at those muscles. Right next to the flexor carpi ulnaris is a very small bellied muscle whose tendon points right at the palm of our hand. What small bellied muscle do you think has a long tendon that points right at the palm of the hand?
Palmaris longus.
There you go. It is indeed the palmaris longus. Right next to that is a tendon for a muscle that goes to the thumb side. And if it is the flexor carpi ulnaris, whose tendon goes to the pinky side in the front, guess what we call the muscle whose tendon goes to the thumb side in the front?
Flexor carpi radialis.
There you go. And lo and behold, after like Phineas Fogg, we have circumnavigated the forearm. We get back to the end. And we're back at the brachioradialis where we began. 3 anterior muscles, 3 posterior muscles. Pay attention to the tendons, right? And they tell you exactly where they are, and their names tell you what you do. Flexors flex, extenders extend. Ulnarises are on the pinky side, medially, so they deduct. Radialises are on the thumb side, so they abduct. And of course, digitalis moves the digits. Palmaris longus goes to the palm, moves the hand. So the names tell you what they do, their tendons tell you what they are. Get that local landmark of the brachioradialis and then circumnavigate the forearm. And it really is just that easy. It's going to take you a little bit of time, but, and again, I promise you we'll go through this again, but I wanted to at least expose you to it before the weekend so that you have that opportunity to study, because this weekend you need to get ahead. Because if you think about it, from today forward, you have 2 more days of lecture and only 3 days to master this material before the next exam. Saturday, Sunday, and Wednesday. And you're still getting new material Monday and Tuesday. So now is the time to get ahead. This is your big weekend to study. All right. Questions on any of that? All right, it's a little bit early, but this is a good stopping point. We got through the forearm, which is what I was really, really hopeful we'd be able to do, and we did that. So excellent. So that is all that I wanted to cover for today. We'll give you guys a head start on the weekend. So I wish you luck. Study hard as always. If you have any questions, feel free to email me. I'm happy to do that. As I've also said, now that you know the 7 big essay questions that you're going to get 2 of, if you want to write a practice version of that and email it to me, I will happily respond and grade it for you to tell you what you got right and what you got wrong and what you can do to improve it. Again, my goal is to help you to be successful, so please take advantage of that. All right, have a good, safe weekend, study hard, and I will see you guys on Monday.
Thank
Thank
you.
you.